Hello and welcome to The Sleepy Scientist. Before settling in, it would be lovely to know where this video is reaching you tonight. Perhaps you're watching from somewhere nearby, or perhaps from the other side of the world. Wherever you happen to be listening from, feel free to share your location in the comments. It is always fascinating to see just how far these quiet little scientific journeys travel. If you enjoy calm explorations of science like this one, please consider subscribing to the channel and giving the video a like. It helps more curious minds discover these slow, thoughtful explanations of how the universe and the human body work. And to everyone who already subscribes and returns regularly for these late-night scientific wanderings, thank you. The continued support makes it possible to keep creating these long, relaxing journeys through science together. Now, tonight's topic is something quietly remarkable. At this very moment, without asking for attention and without announcing itself in any obvious way, an intricate electrical network inside the body is coordinating almost everything happening in life. Muscles move when needed. The heart keeps beating. Breathing continues without instruction. Sensations travel from skin to brain in fractions of a second. Memories form, thoughts appear, decisions happen. All of this is guided by a system so deeply integrated into life that it rarely draws attention to itself. In fact, while listening to these words, the nervous system is already performing an astonishing number of tasks. Electrical signals are traveling through nerves in the fingers holding a phone or resting on a keyboard. Sensory receptors in the ears are converting sound waves into patterns of neural activity. Tiny chemical messengers are passing between brain cells, allowing meaning to emerge from what would otherwise be nothing more than vibrations in the air. And the curious thing is that almost none of this requires conscious effort. The nervous system simply runs in the background, quietly coordinating the body like an invisible operating system. It does this while walking, while sleeping, while daydreaming, and even while paying attention to something entirely different. Yet despite this constant activity, most people rarely pause to wonder how it actually works. How does a network made of microscopic cells manage to control an entire human body? How do electrical signals moving through living tissue become sensations, movement, emotion, and thought? And perhaps most intriguingly of all, how does this silent biological network manage to run nearly every moment of life without ever asking for attention? Tonight, the journey begins inside that hidden network. Inside the human body exists a communication network so extensive, so active, and so quietly reliable that it rarely receives much attention at all. It does not hum like machinery or flash like a computer screen. It does not announce when it begins working each morning or when it finishes at night. In truth, it never stops working. This system is known as the nervous system, and it connects nearly every part of the body in a continuous exchange of information. Signals flow between the brain, the spinal cord, the organs, the muscles, and the countless sensory receptors scattered across the skin and throughout internal tissues. Every moment of life depends on these signals moving back and forth across the body in an organized pattern. The muscles of the chest expand and contract to draw breath. The heart continues its steady rhythm. The eyes adjust to changing light. The stomach releases digestive enzymes when food arrives. Even something as simple as shifting slightly in a chair requires messages to travel from brain to muscle and back again. Most of this happens without any deliberate effort. The nervous system operates largely outside conscious awareness, quietly coordinating the body like an invisible network of communication lines. The brain sends instructions, the body sends reports, and the exchange continues endlessly in both directions. In a sense, the nervous system forms the body's internal conversation. What makes this system particularly fascinating is the way it communicates. Instead of spoken words or written messages, The nervous system uses electrical signals and chemical signals that move through living tissue. Every sensation, every movement, every thought is tied to these patterns of electrical activity travelling along specialised cells known as neurons. The discovery that living tissue could produce electricity once seemed almost unbelievable. During the late 1700s, the Italian scientist Luigi Galvani performed a series of experiments that would change the understanding of biology forever. Galvani was studying the muscles of frogs when he noticed something peculiar. When metal instruments touched the exposed nerves of a frog's leg, the muscles twitched suddenly, even though the animal was no longer alive. At first glance, the phenomenon looked almost magical, as if the metal itself had somehow revived the muscle. Galvani believed he had discovered what he called animal electricity, a natural electrical force produced by living organisms. His experiments suggested that nerves carried electrical signals capable of triggering muscular movement. Another Italian scientist, Alessandro Volta, disagreed with Galvani's interpretation. Volta believed the electricity came from the metals used in the experiment rather than from the animal tissue itself. This disagreement led Volta to develop one of the first electrical batteries, an invention that would transform physics and chemistry. Although the two scientists argued about the source of the electricity, both were partly correct in ways neither fully realized at the time. Living tissue does indeed produce electrical signals, and the study of those signals eventually became known as electrophysiology. Today, electrophysiology is a central field within neuroscience, devoted to understanding how electrical activity moves through neurons and neural circuits. The nervous system, as modern science understands it, operates through a remarkable hierarchy of organization. At the smallest level, the process begins with charged particles known as ions moving across the membranes of cells. Sodium ions, potassium ions, calcium ions, and chloride ions all play roles in generating electrical signals within neurons. These ions move through specialized proteins embedded in the cell membrane called ion channels. When these channels open or close, they alter the electrical balance across the membrane, creating tiny voltage differences between the inside and outside of the cell. When many ion channels open in rapid succession, a wave of electrical change travels along the neuron. This travelling electrical event is known as an action potential. The neuron itself forms the next level of organization. Each neuron has branching structures called dendrites that receive incoming signals, a central region called the cell body, where those signals are processed, and a long projection called the axon that carries electrical impulses away from the cell. Some axons stretch astonishing distances through the body. In the longest nerves, signals can travel from the spinal cord all the way down to the toes. Neurons do not work in isolation. They connect to one another through tiny junctions known as synapses. At these junctions, electrical signals trigger the release of chemical messengers called neurotransmitters. These chemicals cross microscopic gaps between cells and influence the activity of the next neuron in the chain. When billions of neurons connect together in this way, they form circuits. Circuits combine to create larger networks responsible for functions such as movement, sensation, memory, and emotional processing. Entire regions of the brain consist of layered networks interacting continuously with one another. This idea that the nervous system behaves like a network has fascinated scientists for decades. In the 1940s, two researchers named Warren McCulloch and Walter Pitts proposed a bold theory about how neurons might function collectively. They suggested that networks of neurons could perform logical operations similar to those used in mathematics and computing. Their work laid the foundations for computational neuroscience and later inspired the development of artificial neural networks in computer science. Although biological neurons are far more complex than the simplified models used in early theories, the general idea proved surprisingly powerful. Neural systems can indeed be studied as networks that process information through patterns of activity. From this perspective, the nervous system becomes more than a collection of cells. It becomes an information processing system operating inside a living organism. Signals arriving from the senses are interpreted by neural circuits. Decisions are formed through patterns of activity across interconnected regions. Commands are sent to muscles and organs, producing movement and regulating internal processes. Yet even with all the scientific progress made over the past 2 centuries, one profound question still lingers. How do electrical signals travelling through living tissue give rise to subjective experience? A neuron firing in the brain produces a tiny electrical impulse lasting only a fraction of a second. A chemical molecule released at a synapse alters the activity of another neuron. These events are measurable and observable. Instruments can detect them, and scientists can map where they occur. But somewhere within this vast network of electrical and chemical activity emerges the sensation of seeing light, the sound of a voice, the warmth of sunlight on skin, the memory of a childhood moment, or the quiet awareness of being alive. The connection between neural activity and conscious experience remains one of the most intriguing puzzles in science. Some researchers approach the question through neuroscience, studying how patterns of brain activity correlate with perception and thought. Others approach it through philosophy, asking deeper questions about the nature of mind and matter. Regardless of the ultimate explanation, one thing remains clear. The nervous system operates as a continuous control system, governing almost every aspect of human life. It monitors the external world through the senses. It regulates internal organs to maintain stability. It coordinates movement with extraordinary precision. It stores memories, shapes emotions, and allows reasoning to unfold. And it does all of this quietly, rarely asking for attention. At this very moment, signals are racing through nerves at speeds that can reach well over 100 meters per second in some pathways. Thousands of chemical messages are passing between neurons every second. Entire networks within the brain are adjusting their activity as new information arrives from the senses. All of this unfolds continuously, even while the mind focuses on something else entirely. The deeper one looks into the nervous system, the more remarkable it appears. Evolution shaped it gradually over hundreds of millions of years. Physics governs the electrical signals moving through its cells. Chemistry controls the molecules that carry messages between neurons. Biology organizes the structures that connect these signals into functioning networks. Together, these disciplines reveal the nervous system as one of the most intricate systems known in nature. It is the quiet infrastructure of life itself. Long before brains existed, long before animals developed eyes, ears, or limbs, life still faced a basic challenge. A living organism needed some way to sense what was happening around it, and to respond accordingly. Without the ability to detect light, chemicals, temperature, or touch, survival becomes rather difficult. Even the simplest forms of life must react to their surroundings. In the earlier stages of evolution, living organisms solved this problem in extremely simple ways. Single-celled organisms could respond directly to environmental changes through chemical processes inside the cell. A bacterium drifting through water, for example, can detect gradients of nutrients or toxins in its surroundings. By sensing chemical differences, the bacterium adjusts the rotation of tiny structures called flagella, allowing it to swim toward favorable conditions and away from harmful ones. Although bacteria do not possess nervous systems, this behavior demonstrates a foundational principle that would later become central to neural biology. Living systems must detect information from their environment, process that information internally, and generate an appropriate response. As life evolved into multicellular organisms, new challenges appeared. A single cell responding to its environment is relatively straightforward. But when a creature is composed of many cells spread across a body, coordination becomes much more complicated. If one part of the body detects something important, other parts must respond quickly. A signal must travel from one region of the organism to another. This need for coordination eventually gave rise to the earliest forms of nervous systems. Some of the most ancient examples can still be observed today in animals such as jellyfish and hydra. These creatures belong to a group of animals known as cnidarians, which appeared hundreds of millions of years ago. Unlike animals with centralized brains, jellyfish possess what scientists call a nerve net. A nerve net is exactly what the name suggests. Instead of a single central brain controlling the body, neurons are arranged in a loose network distributed throughout the organism. Signals can travel through this network in many directions, allowing different parts of the body to coordinate movement. When a jellyfish encounters prey or a disturbance in the water, sensory cells detect the change and transmit signals through the nerve net. The network activates muscle cells in the bell-shaped body, producing the gentle pulsing motion that propels the jellyfish through the water. No central command center is required. The coordination emerges from the distributed network itself. The hydra, a small freshwater animal related to jellyfish, demonstrates a similar arrangement. Hydra possess a cylindrical body with tentacles surrounding the mouth. Neurons form a network across the body wall, allowing the organism to respond when something touches its tentacles. If a small aquatic animal brushes against the hydra, sensory cells trigger signals through the nerve net, causing the tentacles to contract and capture the prey. These early nervous systems are remarkably simple compared to the human brain, yet they represent a crucial evolutionary innovation. For the first time in the history of life, specialized cells evolved specifically to transmit electrical signals across a multicellular body. Over time, evolutionary pressures encouraged greater coordination and faster responses. As animals began moving more actively through their environments, it became advantageous to concentrate sensory information and decision-making in specific regions of the body. This process eventually led to the emergence of centralized nervous systems. One of the key evolutionary developments associated with centralization is something called bilateral symmetry. Bilateral animals have bodies organized along a left and right axis, with a distinct front and back. Movement typically occurs in the forward direction, meaning that the front of the body encounters environmental information first. Once animals began moving forward through their environments, it became useful to concentrate sensory organs at the leading edge of the body. This phenomenon is known as cephalization. Sensory receptors such as primitive eyes, chemical detectors, and touch-sensitive structures gradually clustered near the front. Neural tissue accumulated there as well, forming the earliest versions of a brain. Instead of signals spreading evenly through a distributed nerve net, sensory information could now travel toward a central processing region. Decisions about movement or behavior could be made more efficiently, and coordinated responses could be sent back through the body. The emergence of centralized nervous systems marked a major turning point in evolutionary history. The idea that the nervous system functions as a coordinated network rather than a simple collection of wires was articulated in remarkable detail by the British physiologist Charles Sherrington during the late 19th and early 20th centuries. Sherrington studied how reflexes operate in animals and how signals travel through the spinal cord and brain. Through careful experiments, he realized that the nervous system does not simply transmit signals from one location to another. Instead, it integrates information from many sources simultaneously. Sensory input from the skin, muscles, and internal organs converges within neural circuits. These circuits process the incoming information, and produce coordinated responses. Sherrington described the nervous system as an integrative system. The word integration here refers to the combination of many signals into a coherent pattern of activity. Rather than functioning like a simple telegraph line, the nervous system behaves more like a network constantly evaluating incoming information and adjusting the body's behavior. Modern neuroscience has continued to build upon this insight. Evolutionary neurobiologist Gabor Jekeli has explored how primitive nervous systems may have evolved to guide movement through environmental sensing. His research examines how early animals use simple sensory systems to orient themselves toward light sources, chemical gradients, or other environmental cues. For example, certain marine larvae possess light-sensitive cells that allow them to detect the direction of incoming light. These cells influence the beating patterns of tiny hair-like structures called cilia, which control the animal's movement through water. By adjusting how the cilia beat, the organism can swim toward the light or away from it. This process demonstrates how sensory information can directly influence movement even in very simple nervous systems. A basic feedback loop emerges. The organism detects a stimulus, processes that information through neural activity, and adjusts its movement accordingly. Over many generations of evolution, these simple feedback systems became more elaborate. The timing of these developments corresponds closely with a major evolutionary event known as the Cambrian diversification, which occurred roughly 540 million years ago. During this period, the fossil record reveals an extraordinary increase in the diversity of animal body plans. Many scientists believe that the rise of more sophisticated nervous systems played a key role in this transformation. As neural circuits became more capable of processing sensory information, animals gained the ability to explore their environments more actively. Predators evolved strategies for detecting and capturing prey, while prey species developed improved sensory systems to detect danger. This evolutionary interaction between predator and prey is sometimes described as an arms race. Improvements in sensory perception, movement, and neural coordination allowed animals to survive in increasingly complex ecosystems. Fossils from Cambrian deposits such as the Burgess Shale in Canada reveal early animals with distinct heads, sensory structures, and neural organization. Creatures like Anomalocaris possessed compound eyes capable of detecting movement in the surrounding water. These eyes likely connected to neural circuits that coordinated rapid swimming and hunting behavior. Such developments demonstrate how environmental complexity can drive the evolution of nervous systems. As ecosystems became richer and more competitive, animals benefited from faster sensory processing, improved coordination, and more efficient control over movement. Neural tissue expanded and specialized, eventually giving rise to the complex brains found in vertebrates. Despite this long evolutionary journey, the fundamental purpose of the nervous system has remained remarkably consistent. At its core, the nervous system exists to detect information, interpret that information, and generate appropriate responses. Whether in a jellyfish drifting through the ocean, a small marine larva orienting towards sunlight, or a human navigating a crowded city street, the underlying principle remains the same. Signals arrive from the environment. Neural circuits process those signals. The body responds. What began hundreds of millions of years ago as a simple network of electrically active cells has gradually evolved into one of the most intricate systems known in biology. And yet, beneath all the complexity, the original purpose remains quietly intact. The nervous system still does what it has always done. It helps life sense the world and respond to it. If the nervous system is a vast communication network, then the true work of that network happens at a much smaller scale. Hidden within the brain, the spinal cord, and the countless nerves that spread throughout the body are tiny living cells designed specifically to transmit information. These cells are known as neurons. A single neuron is not large. Most measure only a small fraction of a millimeter across. Yet the shape of a neuron is immediately distinctive when seen under a microscope. Instead of looking like a simple rounded cell, a neuron extends into long, branching structures that allow it to communicate with many other cells at once. The central portion of the neuron is called the cell body, sometimes referred to as the soma. This region contains the nucleus and most of the internal machinery required to keep the cell alive. From the cell body extend multiple short branches called dendrites. The word dendrite comes from the Greek word for tree, which is quite fitting. Under magnification, these branches spread outward in delicate patterns that resemble the limbs of a small tree. Dendrites serve as receiving structures. Signals arriving from other neurons reach the dendrites first. These incoming messages are then carried toward the cell body, where the neuron evaluates the information it has received. Extending away from the cell body in the opposite direction is a long, slender projection known as the axon. The axon is the neuron's transmitting cable. Electrical signals generated within the neuron travel down this structure and continue toward other cells. Some axons are extremely short, linking neighboring neurons inside the brain. Others can stretch remarkable distances through the body. In the longest nerves, axons extend from the spinal cord all the way down to the muscles of the foot. Even in a tall adult human, a single neuron can span more than a meter in length. Despite that distance, electrical impulses can travel along the axon in a fraction of a second. This arrangement allows each neuron to receive signals through its dendrites, process those signals within the cell body, and then transmit a new signal through the axon. In effect, every neuron functions as a tiny information processing unit embedded within the larger neural network. Today, this structure seems obvious to neuroscientists, but during the 19th century, the true organization of the nervous system was still unclear. Many scientists believed that neural tissue formed a continuous web, a single interconnected network in which signals flowed freely in all directions. The Spanish scientist Santiago Ramón y Cajal changed that understanding completely. Using improved staining techniques developed by the Italian physician Camillo Golgi, Ramón y Cajal spent countless hours examining thin slices of brain tissue under the microscope. The staining method allowed individual neurons to appear dark against a lighter background, revealing their intricate shapes. Where others saw tangled complexity, Ramón y Cajal saw organization. He carefully sketched the neurons he observed, documenting their branching dendrites and long axons in extraordinary detail. His drawings remain famous today for both their scientific precision and their artistic beauty. Through these observations, Ramón y Cajal reached a revolutionary conclusion. The nervous system was not a continuous network. Instead, it was composed of distinct individual cells communicating with one another across small gaps. This idea became known as the neuron doctrine. According to the neuron doctrine, neurons are separate cells that transmit signals in specific directions through organized connections. Each neuron receives input from other neurons, processes that information internally, and then sends signals onward. The neuron doctrine eventually became one of the foundational principles of modern neuroscience. Ramón y Cajal's work was so influential that he shared the Nobel Prize in Physiology or Medicine in 1906 for his discoveries concerning the structure of the nervous system. For a long time, neurons received most of the attention in neuroscience. They were clearly responsible for carrying electrical signals, and they formed the visible wiring of the brain. But neurons are not alone inside neural tissue. Surrounding and supporting these neurons is another vast population of cells known collectively as glial cells. The term glia comes from a Greek word meaning glue. Early researchers believed these cells simply held the nervous system together, acting as a kind of biological scaffolding between neurons. Because of this assumption, glial cells were once considered passive helpers rather than active participants in neural activity. This view began to change through the work of the Spanish neuroscientist Pío del Río Ortega during the early 20th century. Ortega refined microscopic staining techniques and carefully studied the different types of glial cells present in brain tissue. His research led to the identification of two important classes of glial cells known as microglia and oligodendrocytes. Microglia function as the immune cells of the brain. They patrol neural tissue searching for damaged cells, pathogens, or debris that needs to be removed. When injury occurs in the brain, microglia become active and help protect the surrounding neural environment. Oligodendrocytes perform a different role. These cells produce a fatty insulating substance called myelin. Myelin wraps around many axons, forming protective layers that help electrical signals travel more quickly along the nerve fiber. Without this insulation, neural communication would be dramatically slower. For many decades, glial cells were still regarded primarily as support staff for the neurons that carried electrical signals. But modern neuroscience has begun to reveal a more complex picture. Researchers now suspect that glial cells may play active roles in regulating neural communication. Some glial cells can release chemical signals that influence the activity of nearby neurons. Others appear to help shape the strength of synaptic connections between neurons. This emerging field of study has led some scientists to suggest that the brain functions not simply as a network of neurons, but as a cooperative system involving both neurons and glial cells working together. Meanwhile, the organization of neurons into circuits allows the nervous system to perform specialized tasks. The American neuroscientist Joseph Ledoux has studied how specific neural circuits process emotional information, particularly within brain structures involved in fear and threat detection. His research focuses on the amygdala, a small almond-shaped structure deep within the brain. Ledoux demonstrated that sensory signals related to potential danger can travel through dedicated neural pathways that trigger rapid emotional responses. These circuits allow the brain to respond quickly to threats even before conscious reasoning fully evaluates the situation. This discovery highlights an important principle of nervous system organization. Neural circuits are often specialized for particular types of processing. Some circuits handle sensory perception, others coordinate movement, and still others contribute to emotional responses or memory formation. Rather than operating as a single undifferentiated network, the brain contains many interconnected circuits, each performing distinct roles within the overall system. The scale of this network is difficult to imagine. The human brain contains tens of billions of neurons. Each neuron can form connections with thousands of other neurons through synapses. When these connections are counted together, the number of communication pathways in the brain becomes staggering. Estimates suggest that the brain may contain hundreds of trillions of synaptic connections. These connections allow signals to flow through immensely complex patterns of activity that change constantly throughout life. Every memory formed, every movement executed, and every sensation experienced corresponds to patterns of activity within this vast network. Despite the complexity, the basic principle remains beautifully simple. Individual neurons receive information, process it, and transmit signals onward to other neurons. These signals travel through circuits. Circuits combine into networks. Networks coordinate the countless functions required for life. Within this quiet electrical architecture, thoughts arise, sensations are interpreted, and decisions unfold. And all of it begins with a single type of cell designed to carry a signal from one place to another. Deep inside every neuron, an electrical drama is unfolding continuously. It is silent, invisible, and astonishingly small in scale. Yet these tiny electrical changes allow the nervous system to transmit signals across the entire body. The muscles that move the fingers, the receptors that detect sound in the ears, and the brain circuits responsible for memory all rely on this same fundamental process. At the heart of the phenomenon lies a basic principle of biology and physics known as the membrane potential. Every neuron is surrounded by a thin boundary called the cell membrane. This membrane separates the interior of the cell from the fluid environment outside it. Although the membrane is extremely thin, it performs an essential role. It regulates which substances can enter or leave the cell, and in doing so it creates a difference in electrical charge between the inside and the outside of the neuron. The reason this electrical difference exists involves tiny charged particles known as ions. In biological systems, several types of ions play important roles, including sodium ions, potassium ions, calcium ions, and chloride ions. Each of these particles carries a small electrical charge. Inside a resting neuron, potassium ions are relatively abundant, while sodium ions are more concentrated outside the cell. The cell membrane contains proteins that actively maintain this imbalance. One of the most important of these proteins is known as the sodium-potassium pump. This molecular machine uses energy from the cell to move sodium ions out of the neuron and potassium ions into it. As a result of this process, the interior of the neuron becomes slightly more negatively charged compared with the outside. This difference in charge creates the membrane potential. In many neurons, the resting membrane potential is roughly negative 70 millivolts, which means the inside of the cell is 70 thousandths of a volt more negative than the surrounding fluid. Although this voltage difference may sound extremely small, it provides the electrical foundation for neural signaling. The cell membrane is not simply a passive barrier. Embedded within the membrane are specialized proteins known as ion channels. These channels act like microscopic gates that allow specific ions to move across the membrane when certain conditions are met. When a neuron receives signals from other neurons through its dendrites, those signals can cause some ion channels to open. Sodium ions then rush into the cell, temporarily reducing the electrical difference across the membrane. If enough sodium channels open at once, the electrical balance shifts dramatically. The inside of the neuron suddenly becomes more positive than the outside. This rapid change in voltage is called an action potential. Once an action potential begins, it travels along the axon as a moving wave of electrical change. Sodium channels open in sequence along the membrane, allowing the signal to propagate forward. Shortly afterward, potassium channels open to restore the original balance of ions, returning the membrane to its resting state. The entire process unfolds with remarkable speed. In some neurons, action potentials can travel faster than 100 meters per second. Even slower neurons transmit signals quickly enough that communication across the body feels almost instantaneous. The mechanisms behind this electrical behavior were uncovered through a series of groundbreaking experiments conducted during the mid-20th century by two British scientists, Alan Hodgkin and Andrew Huxley. Hodgkin and Huxley were interested in understanding exactly how electrical signals moved along nerve fibers. To investigate the problem, they studied the nervous system of a marine animal known as the squid. Squid possess unusually large nerve fibers called giant axons that control rapid jet propulsion used for escaping predators. These giant axons are much wider than most neurons found in vertebrates, making them easier to study with the instruments available at the time. Hodgkin and Huxley inserted fine electrodes into the squid axons and measured the electrical changes occurring during neural signaling. Through careful experimentation, they discovered that the action potential results from the controlled movement of sodium and potassium ions through voltage-sensitive ion channels in the cell membrane. Sodium ions flow into the neuron during the rising phase of the signal, while potassium ions flow outward during the recovery phase. Their work produced one of the most influential models in neuroscience. The Hodgkin-Huxley model describes how changes in membrane voltage open and close specific ion channels, allowing electrical signals to propagate along neurons. For this research, Hodgkin and Huxley received the Nobel Prize in Physiology or Medicine in 1963. Their discoveries revealed that the nervous system operates according to precise physical and chemical principles. Another scientist who made important contributions to understanding membrane electricity was Kenneth Cole, an American biophysicist whose work helped establish the electrical modeling of cell membranes. Cole developed methods for measuring how electrical currents interact with biological membranes. He demonstrated that cell membranes behave in some ways like electrical circuits, possessing properties similar to resistors and capacitors. These comparisons allowed scientists to apply mathematical models to biological systems, deepening the understanding of how electrical signals move through living cells. Cole's work helped establish electrophysiology as a discipline that bridges biology, physics, and electrical engineering. The neuron could now be studied not only as a biological cell, but also as an electrical system governed by measurable physical laws. This realization is both elegant and slightly surprising. The signals responsible for sensation, movement, and thought obey the same fundamental principles that govern electrical circuits and electrochemical gradients throughout nature. At the same time, biological systems introduce remarkable complexity. Ion channels embedded within neuronal membranes are highly specialized proteins that open and close in response to specific triggers. Some respond to changes in voltage across the membrane. Others open when chemical molecules bind to them. Certain channels allow calcium ions to enter the cell, triggering processes such as neurotransmitter release at synapses. Each neuron contains thousands of these channels arranged across its membrane. Their collective behavior determines how easily the neuron generates electrical signals and how those signals propagate along the axon. The result is a finely tuned electrical system capable of extraordinary responsiveness. A slight change in ion flow can alter how a low neuron responds to incoming information. Small differences in membrane properties can shape how neural circuits behave. Despite the complexity, the overall principle remains consistent throughout the nervous system. Electrical signals travel along neurons in the form of action potentials. These signals move rapidly across neural circuits, allowing different parts of the body to communicate with remarkable efficiency. When a fingertip touches a warm surface, sensory receptors convert the physical stimulus into electrical activity within nearby neurons. Those signals travel through nerves toward the spinal cord and onward to the brain. In response, motor commands may travel back through other neurons to adjust the movement of the hand. All of this happens through the controlled movement of ions across neuronal membranes. In essence, the nervous system speaks an electrical language. Every sensation, every movement, and every thought emerges from patterns of electrical impulses traveling through networks of neurons. What appears from the outside as a quiet moment of thought or a simple physical action is, within the nervous system, a constant choreography of ions, membranes, and electrical signals. And within that silent electrical dance lies the foundation of neural communication. Neurons are remarkable cells, capable of sending electrical signals across surprising distances within the body. Yet even the longest axon eventually comes to an end. When one neuron finishes sending its signal, another neuron must receive it. This handoff does not occur through direct contact. Instead, the signal crosses a tiny gap. That gap is known as a synapse. A synapse is a microscopic junction where one neuron communicates with another. The space separating the two cells is incredibly small, typically only about 20 to 40 nanometers wide. To give some sense of scale, a human hair is roughly 80,000 nanometers across. The synaptic gap is therefore thousands of times thinner than the width of a single hair. Despite its tiny size, the synapse is one of the most important structures in the entire nervous system. It is the place where electrical signals carried along one neuron are translated into chemical messages that influence the activity of the next neuron. When an electrical impulse reaches the end of an axon, it triggers a fascinating sequence of events. The arriving signal opens specialized calcium ion channels located in the membrane at the tip of the neuron. Calcium ions rush into the cell, and their presence causes tiny sacs filled with chemical messengers to move toward the cell membrane. These sacs, known as synaptic vesicles, fuse with the membrane and release their contents into the synaptic gap. The released molecules are called neurotransmitters. Once released, neurotransmitters drift across the tiny synaptic space and bind to receptor proteins on the surface of the neighboring neuron. These receptors respond by opening ion channels or triggering chemical changes inside the receiving cell. In this way, the signal continues its journey through the nervous system. Today, this process seems straightforward, but the discovery that neurons communicate through chemicals rather than direct electrical contact required a remarkable scientific insight. In the early 20th century, scientists debated how signals crossed the gaps between nerve cells. Some researchers believed that electrical signals passed directly from one cell to another. Others suspected that chemicals might be involved. The Austrian pharmacologist Otto Loewi provided decisive evidence through a famous experiment conducted during the early 1920s. According to Loewi's own account, the idea for the experiment came to him in a dream one night. The next morning he rushed to his laboratory and set up a simple but ingenious demonstration using 2 frog hearts. Loewi isolated the heart of a frog while keeping its vagus nerve intact. The vagus nerve is known to slow the heartbeat when stimulated. When Loewi electrically stimulated the vagus nerve, the frog's heart began to beat more slowly. He then collected the fluid surrounding the heart and transferred it to a second frog heart resting in a separate container. Remarkably, the second heart also slowed down, even though its vagus nerve had not been stimulated. This observation revealed something extraordinary. A chemical released by the first heart had travelled through the surrounding fluid and affected the second heart. Loewi called the substance vagus stoph, which simply meant vagus substance. Later research identified the chemical as acetylcholine, one of the most important neurotransmitters in the nervous system. Loewi's experiment provided clear evidence that nerve cells communicate using chemical signals. For this discovery, Otto Loewi later received the Nobel Prize in Physiology or Medicine. Today, scientists know that many different neurotransmitters participate in neural communication. Each plays a slightly different role in shaping how neurons influence one another. Dopamine is one of the most well-known neurotransmitters. It plays important roles in motivation, reward, and movement control. Dopamine signaling is especially important in brain regions involved in reinforcement learning, where the brain evaluates whether certain actions lead to positive outcomes. Serotonin is another influential neurotransmitter, involved in regulating mood, sleep cycles, appetite, and emotional balance. Changes in serotonin signaling have been linked to various mood disorders, which is why many antidepressant medications affect serotonin pathways. Glutamate serves as the primary excitatory neurotransmitter in the brain. When glutamate binds to its receptors, it often increases the likelihood that the receiving neuron will generate an electrical signal. Because of this role, glutamate is deeply involved in processes related to learning and memory. Acetylcholine, the neurotransmitter first demonstrated in Loewi's experiment, plays essential roles in muscle activation and also contributes to attention and memory in the brain. Each of these chemical messengers operates within complex networks of neurons, influencing how signals flow through neural circuits. The synapse itself is not a fixed structure. Its strength can change over time depending on how often it is used. This phenomenon is known as synaptic plasticity. Synaptic plasticity refers to the ability of synapses to strengthen or weaken in response to patterns of neural activity. When two neurons repeatedly activate together, the connection between them often becomes stronger. Signals pass more easily across the synapse, making communication between those neurons more efficient. Conversely, when connections are rarely used, synaptic strength may decrease over time. The brain gradually adjusts its wiring, reinforcing some pathways while allowing others to fade. This process plays a crucial role in learning and memory. When new skills are practiced or new experiences occur, patterns of neural activity reshape the strengths of synaptic connections. Over time, these adjustments allow the nervous system to store information within its network. The concept that chemical signaling plays a central role in brain function has also been explored by neuroscientist Susan Greenfield. Her research has examined how neurochemical activity contributes to the dynamic processes underlying perception, consciousness, and emotional experience. Greenfield has emphasized that the brain's chemical environment is constantly shifting. Neurotransmitter levels rise and fall, receptor sensitivity changes, and networks of neurons adjust their responsiveness over time. Rather than functioning as a rigid machine, the brain behaves more like a fluid system shaped by continuous chemical interaction. When billions of neurons communicate through synapses across the brain and nervous system, the result is an immense web of connections. Each neuron can form thousands of synapses with neighboring cells. These connections create a communication network of staggering complexity. Estimates suggest that the human brain contains hundreds of trillions of synaptic connections. Each connection represents a possible pathway through which information can travel. Within this vast network, electrical signals and chemical messengers work together seamlessly. Electrical impulses race along axons, carrying signals rapidly across long distances. At each synapse, those electrical signals trigger chemical messages that influence the next neuron in the chain. Step by step, signal by signal, this process allows information to flow across the nervous system. What began as a tiny electrical impulse in one neuron becomes part of a vast conversation among billions of cells. When people imagine the brain, the picture that often comes to mind is a soft folded structure resting quietly inside the skull. From the outside, it appears somewhat simple, a greyish mass with curved ridges and grooves. Yet beneath that calm appearance lies one of the most complex biological structures known to science. The human brain contains tens of billions of neurons and an even larger number of glial cells, all arranged into layered structures and interconnected circuits. These cells form networks that allow perception, memory, movement, emotion, and reasoning to occur simultaneously. The brain does not perform one task at a time in isolation. Instead, many different systems operate together, coordinating countless activities every moment of life. One way scientists understand the brain is by examining its major anatomical divisions. Although the brain functions as an integrated whole, certain large regions specialize in particular types of processing. The largest portion is the cerebrum. This is the broad, folded structure that forms the upper part of the brain. Its surface is covered by the cerebral cortex, a thin layer of neural tissue responsible for many of the abilities commonly associated with human cognition. Perception, voluntary movement, language, planning, and complex reasoning all depend heavily on the cerebral cortex. The cortex itself is arranged into two hemispheres, left and right, connected by a thick bundle of nerve fibers known as the corpus callosum. These fibers allow information to travel between the hemispheres, ensuring that the two sides of the brain remain in constant communication. The surface of the cortex appears wrinkled, forming ridges called gyri and grooves known as sulci. These folds increase the surface area of the cortex, allowing more neurons to fit within the limited space inside the skull. If the cortex was spread flat, it would cover an area roughly comparable to a large dinner napkin. Behind the cerebrum lies a smaller but equally important structure called the cerebellum. The name cerebellum comes from Latin and means little brain. Despite its modest size, the cerebellum contains an enormous number of neurons packed into tightly organized layers. The cerebellum plays a central role in coordinating movement and maintaining balance. It receives information from sensory systems and motor regions of the brain, comparing intended movements with the movements actually occurring. By making rapid adjustments, the cerebellum allows actions to become smooth and precise, rather than awkward or unstable. Even something as simple as reaching for a cup of tea requires the cerebellum to continuously fine-tune the motion of muscles in the arm and hand. At the base of the brain sits another crucial region known as the brain stem. The brain stem connects the brain to the spinal cord and regulates many of the body's most fundamental life-sustaining processes. Breathing rhythms, heart rate, swallowing, and certain reflexes are controlled here. The brain stem includes several structures, among them the medulla oblongata, the pons, and the midbrain. Although these areas operate largely outside conscious awareness, their functions are absolutely essential. Damage to the brainstem can disrupt vital processes that keep the body alive. Deep within the brain lie additional structures often referred to collectively as subcortical regions. These include areas such as the thalamus, the hypothalamus, the basal ganglia, and the amygdala. Each plays specialized roles in regulating behavior, emotion, movement, and internal balance. The thalamus acts as a major relay station for sensory information, directing signals from the eyes, ears, and other sensory organs toward appropriate regions of the cerebral cortex. The hypothalamus helps regulate hunger, body temperature, hormone release, and many aspects of internal stability. The basal ganglia contribute to movement control and habit formation, while the amygdala plays an important role in processing emotional signals, particularly those related to fear and threat detection. Understanding how these regions contribute to specific abilities has fascinated scientists for centuries. During the 19th century, physicians began noticing that injuries to certain parts of the brain produced very specific changes in behavior. One of the most influential discoveries came from the French physician Paul Broca. In the 1860s, Broca studied patients who had difficulty producing speech, even though they could still understand language. When these patients later died, Broca examined their brains and noticed damage in a particular region of the left frontal lobe. This region, now called Broca's area, plays a critical role in speech production. Not long afterward, the German neurologist Karl Wernicke described another language-related region located farther back in the temporal lobe. Patients with damage in this area could speak fluently, but their words often lacked clear meaning and they struggled to understand language spoken by others. This region became known as Wernicke's area and is associated with language comprehension. These discoveries helped establish the idea known as functional localization. According to this concept, different regions of the brain perform specialized functions. Certain areas process vision, others control movement, and some participate in language or memory. While functional localization remains an important principle, modern neuroscience has revealed that the brain rarely operates through isolated modules. Instead, most cognitive processes involve distributed networks spanning multiple regions. Language, for example, does not arise solely from Broca's area or Wernicke's area. Instead, it emerges from coordinated activity across several cortical and subcortical regions linked by neural pathways. Research into these networks expanded significantly during the 20th century. One influential contributor was the neuroscientist Patricia Goldman-Rakic, whose work focused on the prefrontal cortex. This region lies near the front of the cerebral cortex and is associated with decision-making, planning, and working memory. Goldman-Rakic demonstrated that neurons within the prefrontal cortex maintain patterns of activity, while information is temporarily held in mind. This ability allows humans to remember a phone number briefly while dialing it, or to keep track of steps in a complex task. Her research showed that cognitive functions such as working memory rely on coordinated activity within specific neural circuits rather than single isolated brain regions. The brain therefore operates as an interconnected system composed of many interacting parts. Each region contributes its own specialized capabilities, yet these parts communicate continuously with one another through vast networks of neurons. Sensory signals arriving from the eyes and ears are processed in visual and auditory regions of the cortex. Motor areas send commands to muscles throughout the body. Emotional centers evaluate the significance of incoming information. Memory systems compare present experiences with past ones. All of these processes occur simultaneously. The result is a brain capable of coordinating perception, movement, reasoning, and internal regulation at the same time. While attention may focus on a particular thought or sensation, countless neural processes continue quietly in the background. This architectural arrangement allows the brain to function not as a collection of separate parts, but as a unified system whose regions cooperate continuously to guide behavior and maintain life. Extending downward from the base of the brain is a structure that quietly carries an enormous share of the nervous system's daily workload. It is long, slender, and well protected inside the vertebral column. Although it rarely receives much attention compared with the brain, life as it is known would not function without it. This structure is the spinal cord. The spinal cord forms the primary communication pathway between the brain and the rest of the body. Every signal that travels from the brain toward muscles, glands, or organs must pass through this narrow column of nervous tissue. At the same time, signals arriving from the skin, joints, and internal organs travel upward through the spinal cord before reaching the brain. In effect, the spinal cord acts as the central highway of neural communication. The cord itself runs from the lower part of the brainstem down through the vertebral column, ending roughly in the lower back. In adults, it measures around 40 to 45 centimeters in length, though exact size varies slightly between individuals. Along its length, the spinal cord is divided into segments that correspond to different regions of the body. From each segment, pairs of spinal nerves emerge and extend outward through small openings between the vertebrae. These nerves branch repeatedly as they travel toward muscles, skin, and internal tissues. Together they form a vast network linking the central nervous system to nearly every part of the body. Within the spinal cord, neural signals travel along organized pathways known as tracts. Some of these tracts carry sensory information upward toward the brain. Others carry motor commands downward from the brain to the body. Ascending sensory pathways begin at receptors scattered throughout the body. When a fingertip touches a warm surface, pressure receptors and temperature receptors in the skin convert that physical stimulus into electrical activity within nearby sensory neurons. The signal travels along a peripheral nerve until it enters the spinal cord. Once inside the spinal cord, the signal continues upward through ascending tracts that carry sensory information toward the brain. These pathways eventually reach higher processing centers where the sensation becomes consciously perceived. Descending motor pathways operate in the opposite direction. Signals originating in motor regions of the brain travel downward through the spinal cord and exit through spinal nerves that lead to muscles. When a person decides to raise a hand, for example, motor commands descend through these pathways to activate the appropriate muscles. This continuous two-way traffic allows the brain and body to remain in constant communication. Yet the spinal cord performs more than simple signal transmission. It also acts as a local processing centre capable of generating rapid responses on its own. One of the most striking examples of this ability appears in spinal reflexes. A reflex is a rapid, automatic response to a stimulus. Consider the familiar experience of touching a hot surface. Almost instantly the hand pulls away, often before there is time to consciously register the heat. This quick reaction occurs because the signal does not need to travel all the way to the brain before a response is produced. Instead, the sensory signal enters the spinal cord and immediately activates a simple neural circuit known as a reflex arc. Within this circuit, sensory neurons communicate with motor neurons through one or more intermediate neurons. The motor neurons then send signals to muscles that withdraw the hand from the source of heat. Only afterward does the information continue upward to the brain, where the sensation of pain becomes fully conscious. This arrangement provides an important protective advantage. By allowing the spinal cord to generate immediate responses, the body can react to potential harm in a fraction of a second. The scientific understanding of sensory and motor pathways owes much to the work of two early 19th century physiologists, Charles Bell and François Magendie. Both researchers investigated how spinal nerves connect to the spinal cord and how these connections influence movement and sensation. Charles Bell, a Scottish anatomist, proposed that certain nerve fibres carried sensory information toward the brain, while others carried motor commands outward to the muscles. Around the same time, the French physiologist François Magendie performed experiments that confirmed this idea. Magendie carefully studied the roots of spinal nerves, where they connect to the spinal cord. Each spinal nerve divides into two branches as it approaches the cord. One branch, known as the dorsal root, enters the back portion of the spinal cord. The other branch, called the ventral root, enters the front. Magendie demonstrated that the dorsal roots carry sensory information into the spinal cord, while the ventral roots carry motor signals outward to muscles. This discovery became known as the Bell-Magendie law and remains a foundational principle in neuroanatomy. Beyond reflexes and signal transmission, the spinal cord also contributes to coordinating posture and movement. Neural circuits within the cord interact with signals arriving from the brain and from sensory receptors in muscles and joints. When a person walks, for instance, the spinal cord helps coordinate the rhythmic patterns of muscle activity required for stepping. These patterns are generated by networks of neurons, sometimes called central pattern generators. Although the brain influences these movements, much of the detailed coordination occurs within the spinal cord itself. The spinal cord also receives continuous feedback from proprioceptors, specialized sensory receptors located in muscles and tendons. These receptors detect changes in muscle length and tension. By monitoring this information, spinal circuits adjust muscle activity to maintain balance and posture. Standing upright, which may feel effortless, actually involves constant adjustments guided by signals flowing through the spinal cord. For this reason, the spinal cord should not be thought of as merely a passive cable, carrying signals between brain and body. Instead, it functions as an active processing center that participates directly in controlling movement and reflex behavior. Signals passing through the cord are modified, integrated, and sometimes redirected by networks of neurons embedded within its grey matter. These internal circuits allow the spinal cord to respond rapidly to sensory input while maintaining communication with higher brain centers. The result is a system capable of both speed and coordination. Messages from the brain travel downward through descending pathways. Sensory information travels upward through ascending pathways. Local circuits generate reflexes and coordinate movement patterns. All of these processes occur continuously within a structure only a few centimeters wide. Though hidden safely inside the spine, the spinal cord quietly manages a remarkable flow of neural activity every moment of life. Leaving the brain and spinal cord, the nervous system spreads outward through the body in every direction. What begins as a central structure inside the skull and spine quickly becomes something far more extensive. Fine threads of nerve tissue branch outward into the neck, the chest, the arms, the abdomen, and the legs. They travel alongside blood vessels, weave through muscles, and reach into the skin and internal organs. This vast extension of neural tissue is known as the peripheral nervous system. The word peripheral simply means outer or surrounding, and in this case it refers to everything in the nervous system that lies outside the brain and spinal cord. While the central nervous system acts as the main processing center, the peripheral nervous system forms the enormous communication network connecting that center to the rest of the body. Through this network, information flows continuously between the body and the brain. Some signals travel inward, carrying sensory information from the environment toward the central nervous system. Other signals travel outward, delivering instructions that control muscles, glands, and organs. Together, these pathways allow the body to sense, react, and move in response to changing conditions. The peripheral nervous system begins with two main groups of nerves. One group arises directly from the brain itself. These are known as the cranial nerves. Cranial nerves emerge from the base of the brain and travel primarily to the head and neck. They play essential roles in vision, hearing, smell, taste, facial movement, and the control of certain internal organs. The optic nerve carries visual information from the eyes to the brain. The auditory nerve transmits sound signals from the inner ear. The facial nerve helps control expressions such as smiling or frowning. Each cranial nerve performs a specific function, yet together they help link the brain to the sensory world of the face and head. The second group of peripheral nerves arises from the spinal cord. These are known as spinal nerves. Along the length of the spinal cord, pairs of spinal nerves exit through openings between the vertebrae. From there they spread outward across the body. Spinal nerves travel into the shoulders, arms, chest, abdomen, and legs, branching repeatedly as they go. Some fibers carry sensory information from the skin, muscles, and joints. Others deliver motor commands that cause muscles to contract and produce movement. The branching pattern of peripheral nerves is often compared to the structure of a tree. A large nerve trunk leaves the spinal cord, then divides into smaller branches. Those branches divide again and again until individual nerve fibers reach their final destinations. A single peripheral nerve may contain thousands of individual nerve fibers bundled together. Within that bundle, some fibers carry sensory signals while others carry motor commands. The mixture of fibers allows a single nerve to transmit information in multiple directions at once. This architecture creates an extraordinary map of neural communication extending throughout the body. Every patch of skin contains sensory receptors connected to nearby nerve endings. When something touches the skin, those receptors convert the physical stimulus into electrical signals that travel along peripheral nerves toward the spinal cord and brain. At the same time, signals from the brain travel outward through other nerve fibers to control muscles. These motor signals allow a person to reach for objects, adjust posture, or step forward while walking. In this way, the peripheral nervous system forms the interface between the internal world of the nervous system and the external world of the environment. The scale of this network is easy to underestimate because most of it remains hidden beneath the skin. Yet taken together, peripheral nerves extend for many thousands of kilometers if all their branches are added together. They form a living wiring system reaching nearly every corner of the body. During the early 20th century, the American neuroanatomist Harold Burr explored an intriguing aspect of electrical activity in living tissue. Burr was interested in whether patterns of electrical fields might exist across entire organisms, not just within individual neurons. Using sensitive measuring instruments, he studied electrical potentials present across different parts of the body. Burr proposed that living organisms might possess organized electrical patterns that guide biological processes such as growth and development. Although some aspects of Burr's ideas remain debated, his work helped draw attention to the broader electrical nature of living systems. Neural activity does not occur in isolation. It is embedded within a complex biological environment where electrical and chemical signals interact across tissues. The peripheral nervous system represents one of the most extensive examples of this electrical communication in action. Consider the simple act of brushing a hand across a wooden table. Sensory receptors in the skin detect pressure, texture, and temperature. These receptors generate electrical impulses in nearby nerve fibers. The signals travel along peripheral nerves toward the spinal cord and onward to the brain. Within fractions of a second, the brain interprets the sensation and may respond by adjusting the movement of the hand. Perhaps the fingers press slightly harder to explore the surface, or perhaps the hand withdraws if the object feels unexpectedly hot. All of this communication occurs through the peripheral nerves. At the same time, these nerves carry information from deep inside the body. Stretch receptors in muscles report changes in length and tension. Internal receptors monitor blood pressure, oxygen levels, and the chemical composition of the bloodstream. Signals from these receptors travel through peripheral nerves toward the central nervous system, allowing internal conditions to be regulated. The peripheral nervous system therefore serves as both messenger and sensor. It gathers information from the environment and the body's internal organs, delivering that information to the brain. It also carries instructions outward so the body can act. Without this network of nerves, the brain would be isolated from the world it's meant to interpret. The remarkable fact is that these nerve pathways extend into almost every region of the body, from the scalp to the soles of the feet, from the eyes and ears to the smallest muscles in the fingers. Peripheral nerves provide a continuous channel of communication. Even areas that seem distant from the brain remain connected through this network. The skin of the back, the muscles of the legs, and the organs deep within the chest all maintain contact with the central nervous system through branching nerve fibers. In this way, the nervous system does not remain confined to the brain alone. It spreads outward, forming a living map of communication that reaches throughout the entire body. Wherever sensation is detected, wherever movement occurs, and wherever the body responds to its surroundings, the peripheral nervous system is quietly at work carrying the signals that make those responses possible. The human body is constantly surrounded by information. Every moment brings subtle changes in pressure, temperature, light, sound, and chemical signals drifting through the air. Yet most of this information never reaches conscious awareness. Instead, it is quietly monitored by an extraordinary network of biological sensors embedded throughout the skin, muscles, joints, and internal organs. These sensors are known as sensory receptors. A sensory receptor is a specialized structure designed to detect a particular type of physical stimulus. When that stimulus appears in the environment, the receptor converts it into electrical signals that can travel through the nervous system. In this way, the body gathers information about the outside world and its own internal state. The process may sound simple, but it represents one of the most remarkable transformations in biology. Physical energy in the environment becomes electrical activity inside living cells. Light becomes a pattern of signals traveling through the optic nerve. Sound becomes electrical impulses within the auditory pathway. The warmth of sunlight on the skin becomes a stream of neural messages flowing toward the brain. Scientists refer to this transformation as sensory transduction. Sensory transduction is the process through which physical energy from the environment is converted into neural activity. Without this process, perception would not exist. The brain itself does not directly experience the outside world. Instead, it receives patterns of electrical signals generated by sensory receptors scattered across the body. These receptors come in many forms, each tuned to detect a particular type of stimulus. One of the most familiar categories is the mechanoreceptors. These receptors respond to mechanical forces such as pressure, vibration, stretching, and movement. Mechanoreceptors are found throughout the skin and within deeper tissues such as muscles and joints. When the skin is touched, the surface slightly deforms. That tiny distortion changes the structure of proteins embedded in the membranes of mechanoreceptor cells. As the proteins shift, ion channels open, allowing charged particles to flow into the cell. This movement of ions alters the electrical state of the cell membrane, generating a signal that can travel along nearby nerve fibers. In this way, the simple act of touching a surface begins with microscopic mechanical changes inside sensory cells. Different mechanoreceptors are tuned to detect different forms of touch. Some respond best to steady pressure, others are especially sensitive to vibration or rapid changes in contact. Still others monitor the stretching of muscles and tendons, helping the body track the position of its limbs. Without these receptors, the body would have no sense of physical contact with the world. Another major class of sensory receptors is the thermoreceptors. These cells detect changes in temperature. They allow the body to distinguish warmth from cold and help maintain safe internal conditions. Thermoreceptors operate through temperature-sensitive proteins embedded within cell membranes. As temperature rises or falls, the structure of these proteins changes slightly. Those structural changes open or close ion channels, producing electrical signals that travel toward the brain. This allows the nervous system to monitor the thermal environment continuously. A cool breeze across the skin, the warmth of sunlight, or the heat of a cup of tea can all be detected through thermoreceptor activity. Closely related to these systems are nociceptors. Nociceptors are sensory receptors specialized for detecting potentially damaging stimuli. They respond to intense pressure, extreme temperatures, and certain chemical signals released during tissue injury. The signals generated by nociceptors are interpreted by the brain as pain. Pain is often experienced as unpleasant, yet it plays a vital protective role. By alerting the nervous system to possible injury, nociceptors encourage rapid withdrawal from harmful situations. If a hand touches a hot surface, nociceptors in the skin immediately detect the excessive heat. Within fractions of a second, electrical signals travel through peripheral nerves to the spinal cord. Reflex pathways can trigger withdrawal of the hand even before the brain becomes fully aware of the pain. In this way, the body protects itself automatically. Behind these sensory abilities lies a remarkable molecular mechanism that remained mysterious for many years. How exactly do cells detect physical forces such as pressure or stretch? For a long time, the proteins responsible for these sensations were unknown. A major breakthrough came through the work of neuroscientist Ardem Patapoutian. Patapoutian and his research team investigated the molecular basis of mechanosensation, the ability to detect mechanical forces. Through careful genetic experiments, they identified a family of ion channels now known as piezo channels. These proteins sit within the membranes of sensory cells and respond directly to mechanical deformation. When pressure is applied to the skin, the membrane of a mechanoreceptor cell changes shape. That mechanical distortion causes piezo channels to open, allowing ions to flow into the cell. The resulting electrical change triggers neural signals that travel toward the brain. The discovery of piezo channels helped explain how the body detects touch At the molecular level, it revealed that the sensation of pressure or vibration begins with tiny structural changes in specialized proteins embedded in cell membranes. This work earned Ardham Pataputian the Nobel Prize in Physiology or Medicine, shared with David Julius, whose research uncovered similar molecular mechanisms involved in temperature sensation. Together, these discoveries demonstrated that sensory perception begins not with large structures in the brain, but with molecular events occurring inside individual cells. At the smallest scale, perception begins with the movement of ions through microscopic channels. From these tiny beginnings, sensory signals grow into complex neural patterns. Once generated in sensory receptors, electrical impulses travel along peripheral nerves toward the spinal cord and brain. Along the way, signals may be modified, amplified, or integrated with other information. The brain then interprets these signals to create the experience of sensation. When a person feels the softness of fabric, the coolness of water, or the vibration of a mobile phone, those experiences arise from patterns of electrical activity flowing through neural circuits. Yet the origins of those signals lie in minute molecular changes within sensory receptors embedded in the body. This perspective reveals something remarkable about perception. The brain never directly touches the outside world. Instead, it interprets signals generated by receptors scattered throughout the body. The feeling of warmth, the pressure of a handshake, or the sting of a cold wind are all constructed from electrical patterns traveling through neural pathways. In other words, the experience of the world begins with microscopic detectors quietly monitoring the environment. Every second, millions of these receptors are active. They track changes in temperature, pressure, chemical signals, and tissue strain. Most of their activity remains unnoticed, quietly informing the nervous system about the state of the body and the environment. Yet without them, perception itself would not exist. The skin would feel nothing. Muscles would lose awareness of their position. Temperature changes would pass unnoticed. Even injuries might occur without warning. Instead, through the coordinated activity of sensory receptors, the nervous system continuously gathers information from the world. These signals form the raw material from which perception, awareness, and experience emerge. Long before the brain forms a conscious thought, the process has already begun at the level of molecules embedded in tiny receptor cells. Light and sound are two of the most familiar features of the human world. A sunrise spreads colour across the sky. A quiet melody drifts through the air. Footsteps echo down a hallway. Yet none of these experiences exist inside the nervous system in their original form. The brain never directly encounters light waves or sound vibrations. Instead, the nervous system converts these physical events into patterns of electrical signals. Vision begins with light entering the eye. Light itself is a form of electromagnetic radiation traveling through space. When it reaches the human eye, it first passes through the transparent front surface called the cornea. The cornea bends incoming light slightly, helping to focus it toward the inner structures of the eye. Behind the cornea lies the pupil, an opening in the center of the colored iris. The iris adjusts the size of the pupil to regulate how much light enters the eye. In bright sunlight, the pupil narrows, reducing the amount of incoming light. In dim conditions, the pupil widens to allow more light to reach the interior. Once inside the eye, light passes through the lens. The lens changes shape slightly depending on whether a person is focusing on nearby objects or distant ones. This process, known as accommodation, ensures that incoming light forms a sharp image on the back surface of the eye. That back surface is the retina. The retina is a thin layer of neural tissue lining the interior of the eye. It contains specialized sensory cells called photoreceptors that detect light. Photoreceptors are responsible for converting light energy into neural signals that can travel through the nervous system. Two main types of photoreceptors exist in the human retina. These are rods and cones. Rod cells are extremely sensitive to low levels of light. They allow humans to see in dim conditions such as twilight or moonlight. However, rods do not provide detailed color information. Instead, they primarily detect brightness and movement. Cone cells operate best in brighter light. They provide the ability to perceive color and fine visual detail. Different types of cone cells respond to different wavelengths of light, corresponding roughly to red, green, and blue regions of the spectrum. When light enters the eye and strikes these photoreceptors, something remarkable happens at the molecular level. Within each photoreceptor cell are light-sensitive proteins known as photopigments. When photons of light strike these molecules, their structure changes slightly. That structural change triggers a cascade of chemical reactions inside the cell. These reactions alter the electrical state of the photoreceptor membrane. As a result, electrical signals begin to flow through connected neurons in the retina. The signals are gradually processed by layers of retinal neurons before leaving the eye through the optic nerve. The optic nerve carries these signals toward the brain, where they eventually reach a region known as the visual cortex. The interpretation of visual information within the brain has fascinated scientists for centuries. During the 19th century, the German physiologist Hermann von Helmholtz made foundational contributions to understanding how sensory systems function. Helmholtz studied both vision and hearing, helping to establish the scientific field of sensory physiology. He proposed that perception involves active interpretation by the brain, rather than simple recording of sensory input. According to Helmholtz, the brain continuously constructs a representation of the world based on incoming signals and prior experience. This idea helped shift scientific thinking away from the notion that the senses simply mirror reality. Instead, perception was understood as an active, biological process. During the 20th century, new experiments revealed how visual information is organized within the brain itself. Two neuroscientists, David Hubel and Torsten Wiesel, conducted groundbreaking research on the visual cortex. Working with animal models, they recorded electrical activity from neurons within the visual cortex while presenting different visual patterns. Their experiments revealed that many neurons respond selectively to specific visual features such as edges, orientations, and movement. Some neurons fire strongly when a line appears at a particular angle. Others respond when objects move across the visual field. Still others detect contrast between light and dark regions. These findings demonstrated that the brain processes visual information through layers of specialized neurons. Rather than simply displaying an image like a camera screen, the brain extracts patterns and features from incoming signals. From these patterns, the brain constructs the experience of seeing. A similar transformation occurs in the auditory system. Sound begins as vibrations in the air. When an object moves or vibrates, it pushes against nearby air molecules, creating waves of pressure that travel outward. These waves eventually reach the ear. The outer ear, including the visible structure called the pinna, helps gather sound waves and guide them into the ear canal. The waves then reach the eardrum, a thin membrane that vibrates in response to incoming pressure changes. Behind the eardrum lies a small chamber containing 3 tiny bones known as the ossicles. These bones, called the malleus, incus, and stapes, transmit vibrations from the eardrum toward the inner ear while slightly amplifying the signal. The vibrations eventually reach the cochlea. The cochlea is a spiral-shaped structure deep within the inner ear. It is filled with fluid and lined with thousands of delicate sensory cells known as hair cells. These hair cells are the key detectors responsible for translating sound vibrations into neural signals. As vibrations enter the cochlea, they create waves within the fluid inside. These waves cause specific regions of the cochlea to move depending on the frequency of the sound. High-frequency sounds activate one region, while lower-frequency sounds activate another. The movement of fluid bends tiny hair-like structures extending from the tops of the hair cells. When these structures bend, ion channels open in the cell membranes, altering the electrical state of the cells. This electrical change generates signals that travel through the auditory nerve toward the brain. Inside the brain, these signals are analyzed within the auditory cortex. There, neurons interpret patterns related to pitch, rhythm, and intensity. From these patterns emerges the perception of sound. The voice of a friend, the rustling of leaves, or the distant rumble of thunder all begin as mechanical vibrations in the air, yet by the time the brain processes them, they have become structured neural signals interpreted as meaningful experiences. What vision and hearing share is the transformation of physical energy into patterns of neural activity. Light waves striking photoreceptors in the retina and sound vibrations bending hair cells in the cochlea both trigger electrical signals that travel through neural pathways. These signals are then organized and interpreted by specialized regions of the brain. From these electrical patterns arises the vivid sensory world that people experience every day. It is easy to forget that what appears as color, shape, music, and speech is ultimately the product of neural activity. The nervous system continuously translates external events into electrical language, allowing the brain to construct a model of the surrounding environment. Through this process, light becomes sight and vibration becomes sound. The human body is capable of an astonishing variety of movements. Fingers type across a keyboard. Legs adjust their stride as the ground changes beneath them. Eyes shift rapidly from one point of interest to another. Even the quiet act of breathing requires carefully coordinated muscular activity. Behind each of these movements lies a chain of communication running through the nervous system. At the center of this process are motor neurons. These specialized nerve cells carry signals from the central nervous system outward to muscles. While sensory neurons carry information inward from the environment, motor neurons carry instructions outward, telling muscles when to contract and how strongly to do so. Every voluntary movement begins with activity in the brain. Within the outer layer of the brain, known as the cerebral cortex, there exists a region called the motor cortex. This area lies roughly along the top and sides of the brain and plays a crucial role in planning and initiating voluntary movement. When a person decides to lift a hand or take a step forward, networks of neurons within the motor cortex become active. These neurons send signals downward through pathways that travel through the brainstem and into the spinal cord. From the spinal cord, motor neurons carry the instructions outward through peripheral nerves until they reach the appropriate muscles. The motor cortex itself is organized in a remarkable way. Different regions correspond to different parts of the body. A section near the top of the motor cortex controls movement of the legs. Regions slightly lower help control the arms and hands. Even smaller regions are dedicated to fine movements of the face, lips, and tongue. Scientists often illustrate this arrangement using what is sometimes called the motor homunculus. The term homunculus simply means little human figure. In this representation, body parts that require fine motor control, such as the hands and face, appear greatly enlarged because they occupy larger areas of the motor cortex. This reflects an important principle of neural organization. The more precise and delicate a movement must be, the more neural resources are devoted to controlling it. However, the motor cortex does not operate alone. Movement is refined and coordinated through the work of another major structure known as the cerebellum. The cerebellum sits beneath the main cerebral hemispheres at the back of the brain. Although smaller in size than the cerebrum, it contains an extraordinarily dense population of neurons. The cerebellum plays a critical role in maintaining balance, refining motion, and coordinating complex sequences of movement. When the brain issues a command for a movement, the cerebellum compares that command with incoming sensory information from the muscles and joints. If the movement begins to deviate from the intended path, the cerebellum helps adjust the signal to keep the motion smooth and accurate. For example, when reaching for a cup on a table, the motor cortex sends signals initiating the movement of the arm. As the arm moves, sensory receptors within the muscles report information about position and tension. The cerebellum processes this feedback and helps fine-tune the motion so the hand approaches the cup accurately. Without the cerebellum, movements would become clumsy and poorly coordinated. Individuals with cerebellar damage often experience difficulties with balance, precision, and timing. Once signals from the brain travel through the spinal cord and peripheral nerves, they eventually reach their final destination at the muscles themselves. This connection occurs at a specialized structure known as the neuromuscular junction. The neuromuscular junction is the tiny interface where a motor neuron communicates with a muscle fiber. Although it operates on a microscopic scale, it is responsible for translating neural commands into physical movement. When an electrical signal arrives at the end of a motor neuron, it triggers the release of a chemical messenger called acetylcholine. Molecules of acetylcholine are released into the small gap between the nerve ending and the muscle cell membrane. These molecules bind to receptors on the muscle cell, causing ion channels to open. As ions flow into the muscle cell, the electrical state of the membrane changes. This electrical change triggers a cascade of events inside the muscle fiber, ultimately causing the muscle to contract. In this way, an electrical signal traveling through a neuron becomes a mechanical movement within a muscle. The entire sequence occurs with extraordinary speed. Signals can travel along motor neurons at speeds reaching many tens of meters per second. Within fractions of a second, the brain can send commands that lead to coordinated muscle contractions across the body. Yet movement is rarely as simple as activating a single muscle. Most actions require the coordinated activity of many muscles working together. Some muscles contract while others relax. Some stabilize joints while others generate motion. This complexity led scientists to investigate how the nervous system manages such intricate coordination. One influential researcher who explored this question was the Russian physiologist Nikolai Bernstein. Bernstein studied human movement in detail during the early 20th century. By carefully analyzing motions such as walking, hammering, and writing, he observed that the body must solve what he called the problem of motor redundancy. The human body contains far more muscles and joints than would be required for any single movement. For example, there are countless ways to move the arm to reach a cup on a table, yet the nervous system consistently produces smooth, efficient actions. Bernstein proposed that the brain does not control each muscle individually in isolation. Instead, it organizes groups of muscles into coordinated patterns known as motor synergies. A motor synergy is a pattern of muscle activation that works together to produce a particular movement. By activating these patterns, the nervous system can simplify the complex task of controlling many muscles simultaneously. This concept helped shift scientific thinking about motor control. Rather than issuing detailed commands to every individual muscle fiber, the nervous system may organize movement through flexible patterns of coordination. Modern neuroscience continues to explore this idea through research in motor control, robotics, and computational neuroscience. Another remarkable feature of the motor system is its ability to learn. When a person practices a skill such as playing a musical instrument or riding a bicycle, neural circuits gradually adapt. Repeated practice strengthens certain pathways within the motor system while refining the timing and coordination of muscle activity. Over time, movements that once required conscious attention become automatic. This ability to refine movement reflects the dynamic nature of the nervous system. Motor circuits are not fixed or rigid. They adapt through experience, allowing humans to develop complex physical abilities ranging from delicate finger movements to powerful athletic actions. Yet throughout all of this activity, the nervous system operates quietly behind the scenes. Every step taken while walking, every word spoken during conversation, and every gesture made with the hands depends on signals flowing through motor neurons across neuromuscular junctions and into contracting muscle fibers. Most of the time, this entire system works without conscious awareness. The decision to move may begin as a simple intention within the mind, but the execution of that movement involves an intricate collaboration between the motor cortex, the cerebellum, the spinal cord, peripheral nerves, and the muscles themselves. Together they form one of the most elegant systems of coordination found in biology. Through this silent chain of communication, the nervous system transforms thought into action. Not every process in the body waits for conscious approval. The heart continues to beat whether attention is directed toward it or not. The lungs quietly adjust breathing during sleep. Blood vessels narrow or widen as conditions change. The digestive system keeps moving food through the body without requiring a single deliberate command. Behind these quiet adjustments lies a specialized branch of the nervous system, known as the autonomic nervous system. The word autonomic refers to automatic control. This part of the nervous system regulates many internal processes that must continue operating even when the mind is focused elsewhere. It helps maintain internal stability while the body encounters changing circumstances. The autonomic nervous system monitors and regulates heart rate, blood pressure, digestion, pupil size, gland activity, and many other functions necessary for survival. Most of these processes occur beneath the level of conscious awareness. A person rarely needs to think about adjusting their blood pressure or coordinating digestive enzymes. The nervous system manages these tasks continuously in the background. This quiet control system operates through two major divisions that work together in a delicate balance. One division is known as the sympathetic nervous system. The other is called the parasympathetic nervous system. The sympathetic nervous system is often associated with activation and readiness. When the body detects a challenge or a potential threat, sympathetic activity increases. Heart rate rises. Breathing becomes deeper and faster. Blood vessels redirect circulation toward muscles that may be needed for rapid movement. In contrast, the parasympathetic nervous system supports rest, recovery, and maintenance. When parasympathetic activity dominates, the heart rate slows, digestion becomes more active, and the body enters a calmer physiological state. Together, these two systems create a dynamic balance that adjusts bodily functions moment by moment. During the early 20th century, the American physiologist Walter Cannon studied how the body responds to danger and stress. Through experiments and careful observation, Cannon described what he called the fight or flight response. This response is a coordinated pattern of physiological changes triggered by sympathetic nervous system activity. When an animal perceives a threat, the body rapidly prepares either to confront the danger or to escape from it. Heart rate increases to deliver oxygen to muscles. Breathing accelerates to support increased metabolic demand. Pupils widen to allow more light into the eyes, improving visual awareness. Blood flow shifts away from the digestive system and towards skeletal muscles. Cannon recognized that these changes occur quickly and automatically, allowing animals to react to threats without needing to consciously plan every detail. This response is deeply rooted in evolutionary history. For early mammals navigating environments filled with predators and uncertain conditions, rapid physiological adjustments could mean the difference between survival and danger. Even in modern life, the same biological machinery remains active. A sudden loud noise, a stressful situation, or even an intense emotional experience can activate sympathetic pathways that prepare the body for action. Once the challenge passes, the parasympathetic nervous system helps restore balance. Heart rate gradually slows, breathing becomes calmer, digestion resumes its normal rhythm. The shifting balance between sympathetic and parasympathetic activity helps maintain what physiologists call homeostasis. Homeostasis refers to the tendency of living systems to regulate internal conditions within stable ranges despite changing external environments. In other words, the body constantly works to keep its internal environment stable. A major component of this regulatory system is a nerve known as the vagus nerve. The vagus nerve is one of the longest cranial nerves in the body. It begins in the brainstem and extends downward through the neck and chest into the abdomen, connecting the brain with many internal organs. The vagus nerve carries signals that help regulate heart rate, digestion, immune responses, and other bodily functions. Because of its wide reach, it plays a central role in parasympathetic control. In recent decades, neuroscientist Stephen Porges proposed a framework called the polyvagal theory to explore how the vagus nerve may influence emotional and physiological states. Polyvagal theory suggests that the vagus nerve contains multiple pathways that evolved at different stages in vertebrate history. According to this framework, certain branches of the vagus nerve help regulate calm social engagement, while others may contribute to defensive physiological responses during stress. Porges proposed that these different neural circuits help explain why the body can shift between states of relaxation, alertness, and defensive withdrawal, depending on circumstances. While aspects of polyvagal theory remain actively discussed and evaluated within the scientific community, the broader idea that vagus nerve activity influences emotional regulation and physiological balance has inspired considerable research. Scientists continue to study how vagal signals affect heart rhythm, inflammation, digestion, and even aspects of mood. What makes the autonomic nervous system particularly remarkable is how much of its activity occurs without conscious direction. A person does not decide to release digestive enzymes after a meal. The body detects nutrients entering the stomach and intestines, triggering neural and hormonal signals that coordinate digestion. Similarly, breathing patterns adjust automatically when the body exercises or rests. Blood vessels constrict or dilate depending on temperature, physical activity, and emotional states. These processes occur through intricate communication between the brainstem, the hypothalamus, peripheral nerves, and internal organs. The hypothalamus plays a central role in coordinating autonomic regulation. Located deep within the brain, the hypothalamus integrates signals from many sources and helps control body temperature, hunger, thirst, and hormone release. Through its connections with the autonomic nervous system, the hypothalamus helps maintain internal balance while responding to environmental conditions. For example, when body temperature rises during exercise, the hypothalamus activates pathways that increase sweat production and widen blood vessels in the skin. These changes help release excess heat and prevent overheating. Such adjustments occur seamlessly without requiring deliberate attention. Even emotional experiences influence autonomic activity. Anxiety may accelerate the heart and tighten muscles. Calm environments may encourage slower breathing and reduced heart rate. These shifts illustrate how closely the autonomic nervous system links physiological regulation with emotional states. Despite operating largely outside conscious awareness, the autonomic nervous system is essential for survival. It ensures that vital processes continue even during sleep or unconsciousness. Every moment, signals travel through autonomic pathways regulating the rhythm of the heart, the pace of breathing, the movement of the digestive tract, and the balance of internal chemistry. The body, therefore, contains a control system that functions quietly and continuously, adjusting internal conditions with remarkable precision. While conscious thought often receives the spotlight when discussing the nervous system, much of the body's daily operation unfolds beneath awareness. The heart beats, lungs breathe, and organs perform their tasks through neural circuits that require no permission from the conscious mind. This hidden network of regulation allows the body to maintain stability while the mind focuses on the wider world. The human body lives in a constantly changing world. Temperatures rise and fall, food becomes available or scarce, activity levels shift throughout the day, yet inside the body, many conditions must remain remarkably stable if life is to continue smoothly. Body temperature must stay within a narrow range. The concentration of salts and sugars in the blood must remain balanced. Cells require steady supplies of oxygen, nutrients, and chemical signals. Maintaining this internal stability is one of the most important tasks the nervous system performs. During the 19th century, the French physiologist Claude Bernard introduced a powerful idea that helped explain how living organisms survive in changing environments. Bernard proposed that the body maintains what he called the internal environment, a stable internal condition that allows cells to function properly regardless of external fluctuations. Bernard argued that the stability of this internal environment is essential for life. Even if the world outside the body changes dramatically, the conditions surrounding the body's cells must remain carefully regulated. Later scientists built upon Bernard's insight and developed the concept known as homeostasis. Homeostasis refers to the ability of living organisms to maintain relatively constant internal conditions despite changing external circumstances. The word itself combines ideas of stability and balance. Rather than remaining perfectly fixed, internal conditions fluctuate gently around stable ranges that support normal biological function. The nervous system plays a central role in maintaining this balance. At the center of this regulatory network lies a small but extraordinarily important region of the brain called the hypothalamus. The hypothalamus sits deep within the brain near its base, positioned just above the pituitary gland. Although small in size, it functions as one of the body's most important control centers. The hypothalamus continuously monitors signals from the body's internal environment. It receives information from sensors that track body temperature, blood chemistry, hydration levels, and many other physiological conditions. When the hypothalamus detects a change in these conditions, it initiates responses that help restore balance. Consider body temperature as an example. Human cells operate best within a narrow temperature range close to 37 degrees Celsius. Even small deviations from this range can disrupt enzyme activity and cellular processes. Specialized temperature-sensitive neurons within the hypothalamus monitor both internal blood temperature and signals from temperature receptors in the skin. If body temperature rises too high, the hypothalamus activates mechanisms that help release heat. Blood vessels in the skin widen, increasing heat loss. Sweat glands become active, producing moisture that cools the body as it evaporates. These responses occur automatically without requiring conscious attention. If body temperature falls too low, the hypothalamus triggers different responses. Blood vessels in the skin constrict to conserve heat. Muscles may begin to shiver, generating warmth through rapid contractions. Through these adjustments, the body maintains a stable internal temperature even as environmental conditions change. The hypothalamus also monitors hunger and thirst. Cells throughout the body require a continuous supply of nutrients and water to function properly. When blood glucose levels begin to fall, or when the body becomes dehydrated, sensors send signals to the hypothalamus indicating the need for food or fluid. The hypothalamus then activates neural pathways and hormonal signals that generate sensations of hunger or thirst. These sensations encourage behavior that restores balance by seeking food or water. Although hunger and thirst feel like conscious experiences, they originate from the quiet regulatory work of neural circuits deep within the brain. Another remarkable feature of the hypothalamus is its close connection to the endocrine system. The endocrine system consists of glands that release hormones into the bloodstream. Hormones are chemical messengers that travel throughout the body, influencing the activity of distant cells and organs. While the nervous system communicates rapidly through electrical signals along nerve fibers, the endocrine system communicates more slowly through hormones circulating in the blood. The hypothalamus forms a bridge between these two communication systems. This connection occurs through a process known as neuroendocrine signaling. In neuroendocrine signaling, neurons in the hypothalamus release chemical signals that influence hormone secretion from the pituitary gland. The pituitary gland sits directly beneath the hypothalamus and is often described as the master gland of the endocrine system. It releases hormones that regulate the activity of other endocrine glands throughout the body, including the thyroid, adrenal, glands, and reproductive organs. Through this connection, the hypothalamus can influence a wide range of physiological processes, including metabolism, growth, reproduction, and stress responses. A major advance in understanding this relationship came through the work of the British physiologist Geoffrey Harris. During the 20th century, Harris conducted experiments exploring how the hypothalamus communicates with the pituitary gland. His research demonstrated that the hypothalamus releases chemical factors that travel through specialized blood vessels to regulate pituitary hormone secretion. Harris showed that these hypothalamic signals control the release of hormones that influence many bodily systems. His work helped establish the modern understanding of hypothalamic control over endocrine activity. This discovery revealed that the nervous system and endocrine system are not separate regulatory networks. Instead, they form an integrated system that coordinates physiology across the entire body. When stress occurs, for example, the hypothalamus activates neural pathways that stimulate the sympathetic nervous system, while also triggering hormonal signals that influence the adrenal glands. Similarly, when energy supplies run low, hypothalamic circuits influence both appetite-related behavior and hormonal signals that adjust metabolism. Through these mechanisms, the nervous system monitors internal conditions and initiates responses that help maintain stability. Yet the scale of this monitoring is astonishing. Sensors throughout the body continuously report information about oxygen levels, blood pressure, chemical composition of the bloodstream, and many other variables. The nervous system integrates these signals and adjusts bodily functions accordingly. Breathing patterns change as oxygen demand shifts. Heart rate rises during physical exertion and slows during rest. Hormone release fluctuates across the day in rhythms that coordinate sleep, metabolism, and alertness. Much of this activity occurs quietly beneath conscious awareness. Most people rarely think about the fact that their internal chemistry is being monitored and adjusted every moment. Yet without this constant regulation, life would quickly become unstable. Cells depend on steady conditions to carry out the complex reactions required for metabolism, repair, and growth. Homeostasis therefore represents one of the most remarkable achievements of biological organization. Through the combined activity of neural circuits, hormonal signals, and feedback systems, the body maintains an internal environment that allows its trillions of cells to function properly. The nervous system sits at the center of this regulatory network, continuously receiving information comparing conditions to stable ranges and initiating adjustments when necessary. While conscious thought often draws attention, the quiet regulation of internal balance remains one of the most important roles the nervous system performs. It is a silent form of intelligence working constantly in the background, ensuring that life continues smoothly from moment to moment. The nervous system is not a rigid machine assembled once and left unchanged. Instead, it is a living network that constantly reshapes itself. Every new experience, every skill practiced, and every memory formed leaves subtle traces within the structure of neural circuits. This remarkable capacity for change is known as neuroplasticity. Neuroplasticity refers to the ability of the brain and nervous system to modify their connections and patterns of activity in response to experience. Rather than remaining fixed, neural circuits adapt as the organism learns, remembers, and interacts with the environment. For much of scientific history, many researchers believed that the adult brain was largely unchangeable. It was once widely assumed that neural connections were mostly set early in life and remained stable afterward. However, research throughout the 20th century gradually overturned that assumption. Evidence began to show that neural connections could strengthen, weaken, or reorganize depending on how frequently they were used. One of the most influential ideas in this area came from the Canadian psychologist Donald Hebb. In the middle of the 20th century, Hebb proposed a principle that would later become a cornerstone of modern neuroscience. Hebb suggested that when one neuron repeatedly contributes to the activation of another neuron, the connection between them becomes stronger over time. This principle is often summarized in a simple phrase, sometimes described as neurons that fire together tend to wire together. Although the phrase itself is a later simplification, the underlying idea captures Hebb's proposal. Repeated patterns of activity within neural circuits gradually reinforce the connections between participating neurons. When two neurons frequently activate together, chemical and structural changes occur at the synapse connecting them. These changes make future communication between those neurons more efficient. Through this process, patterns of neural activity become easier to trigger again later. This forms one of the biological foundations of learning and memory. To understand how these changes occur at the cellular level, scientists turn to detailed experimental studies of synaptic communication. One of the most important contributors to this work was the neuroscientist Eric Kandel. Kandel studied the biological basis of memory using a simple marine organism called Aplysia, a type of sea slug. Although the nervous system of Aplysia is much simpler than that of humans, its neurons are large and relatively easy to study. Kandel and his colleagues discovered that learning could alter the strength of synaptic connections between neurons. When certain neural pathways were repeatedly activated during learning tasks, the synapses connecting those neurons became stronger. These changes involved modifications in neurotransmitter release, alterations in receptor sensitivity, and even structural changes in the shape of synaptic connections. Kandel's research demonstrated that memory formation involves measurable biological changes within neural circuits. Learning does not simply store information in an abstract way. Instead, it reshapes the physical connections between neurons. This insight helped transform the scientific understanding of memory from a mysterious mental process into a biological phenomenon grounded in cellular mechanisms. Another major discovery in this field emerged from experiments conducted by the Norwegian neuroscientist Terje Lumo and the British neuroscientist Timothy Bliss during the 1970s. While studying the hippocampus, a brain region strongly associated with memory, Lumo observed an intriguing effect. When certain neural pathways were stimulated with rapid bursts of electrical activity, the strength of synaptic communication between those neurons increased dramatically. Even more remarkable was the fact that this increase in synaptic strength could persist for long periods of time after the initial stimulation. Bliss and Lumo carefully studied this phenomenon and gave it a name that has since become central to neuroscience. They called it long-term potentiation. Long-term potentiation refers to a long-lasting increase in synaptic strength following repeated activation of a neural pathway. In practical terms, it means that once a connection between two neurons has been strongly activated, it becomes easier for those neurons to communicate in the future. This process is now considered one of the key cellular mechanisms underlying learning and memory. At the molecular level, long-term potentiation involves changes in the number and sensitivity of neurotransmitter receptors located at synapses. When a synapse is repeatedly activated, additional receptors may be inserted into the receiving neuron's membrane, making it more responsive to future signals. Structural changes can also occur. The tiny protrusions on neurons known as dendritic spines may grow larger or form new connections, increasing the efficiency of communication between cells. Over time, these small modifications accumulate across neural networks throughout the brain. The result is that experiences gradually reshape the architecture of neural circuits. When someone learns to play a musical instrument, the repeated practice of specific movements strengthens neural pathways controlling those motions. When a language is learned, patterns of neural activity associated with vocabulary and grammar become increasingly refined. Even everyday experiences such as navigating a familiar street or recognizing a friend's voice contribute to the gradual shaping of neural circuits. Neuroplasticity does not occur only during childhood. Although the developing brain shows particularly high levels of plasticity, the adult brain retains the ability to change throughout life. Studies of individuals recovering from brain injury have revealed how surviving neural circuits can reorganize to compensate for damaged areas. In some cases, functions that were once performed by one region of the brain can be partially taken over by other regions. Similarly, individuals who lose a sense such as vision may develop enhanced abilities in other sensory systems. This occurs because neural circuits reorganize to process information in new ways. These observations illustrate the remarkable adaptability of the nervous system. Even at rest, neural circuits remain dynamic. Synapses are continuously strengthened, weakened, created, or eliminated depending on patterns of activity. This ongoing process allows the brain to refine its internal models of the world. Learning, therefore, represents not a single event, but an ongoing biological process unfolding within billions of interconnected neurons. Every new experience leaves traces within the nervous system. Some of these traces are temporary, fading quickly if they are not reinforced. Others become stable patterns of connectivity that persist for years. From the perspective of neural circuits, memory is not stored in a single location. Instead, it exists as distributed patterns of strengthened connections spread across networks of neurons. Through neuroplasticity, the nervous system remains flexible enough to adapt to new challenges, while retaining knowledge from past experiences. The quiet rewiring of neural connections continues throughout life, shaping perception, behavior, and skill. While much of this process occurs outside conscious awareness, it allows the brain to remain a living system capable of learning from the world it inhabits. In this sense, every thought, every action, and every experience subtly reshapes the neural landscape within the brain. Within the brain, thoughts and emotions do not arise from a single isolated structure. Instead, they emerge from networks of interconnected regions that communicate constantly through electrical and chemical signals. Some of these networks are especially important for processing emotional experience, forming memories, and shaping behavior in response to the world. One of the most important of these systems is known as the limbic system. The limbic system refers to a group of structures located deep within the brain that play key roles in emotion, motivation, and memory. Although the exact boundaries of the limbic system remain a topic of scientific discussion, several core regions are consistently included in this network. Among these are the hippocampus, the amygdala, and parts of the hypothalamus and surrounding cortical areas. These structures form interconnected circuits that influence how experiences are remembered, how threats are detected, and how emotional responses are generated. One of the central components of this network is the hippocampus. The name hippocampus comes from a Greek word meaning seahorse, reflecting the curved shape of this structure when viewed in cross-section. The hippocampus plays a crucial role in forming new memories. When experiences occur, patterns of neural activity representing those experiences pass through the hippocampus. The hippocampus helps organize and consolidate these patterns so they can be stored more permanently across other regions of the brain. Without a functioning hippocampus, the ability to form new long-term memories becomes severely impaired. This became dramatically clear through the study of a patient known in scientific literature as Henry Molaison. After surgery removed large portions of his hippocampus in the mid-20th century, Molaison retained memories from earlier in life but lost the ability to form new long-term memories. His case provided powerful evidence that the hippocampus is essential for transforming short-term experiences into lasting memories, yet memory is not the only process shaped by limbic circuits. Another key structure within this system is the amygdala. The amygdala consists of small almond-shaped clusters of neurons located deep within the temporal lobes of the brain. The amygdala plays an important role in detecting emotionally significant events, particularly those related to potential threats. When the brain receives sensory information suggesting danger, the amygdala can rapidly activate responses that prepare the body for action. These responses may include increased heart rate, heightened attention, and the release of stress-related hormones. The amygdala also contributes to emotional learning. Experiences that involve strong emotional reactions often become deeply memorable. This occurs because the amygdala interacts with memory-related structures such as the hippocampus, strengthening the encoding of emotionally important events. For example, an unexpected loud noise may create a lasting memory not only because of the sensory experience, but also because the amygdala signals that the event may have been significant for survival. Through these interactions, emotional systems help shape what the brain chooses to remember. Understanding how emotional processes operate within the brain became a major focus of research during the late 20th century. One of the scientists who made influential contributions in this field was the neuroscientist Jaak Panksepp. Panksepp helped establish a field known as affective neuroscience, which studies the neural mechanisms underlying emotional experiences. Through experiments involving both animals and humans, Panksepp proposed that several fundamental emotional systems are deeply embedded within mammalian brains. These systems appear to generate basic emotional states that guide behavior. Among the emotional systems Panksepp described were networks related to seeking, fear, rage, care, and play. The seeking system, for example, motivates exploration and goal-directed behavior. It encourages animals to investigate their surroundings and search for resources such as food or shelter. The fear system helps organisms respond to potential threats by activating defensive behaviors. The care system supports nurturing behaviors that promote the survival of offspring. Panksepp suggested that these emotional systems evolved early in mammalian history and remain deeply rooted within neural circuits shared by many species. This perspective highlights an important principle. Emotions are not merely abstract experiences shaped by culture or language. They are grounded in biological systems that evolved to help organisms survive and reproduce. Another layer of emotional regulation involves the influence of neurotransmitters. Neurotransmitters are chemical messengers released by neurons to communicate with other cells. different neurotransmitter systems influence mood, motivation, and emotional tone. One example is dopamine, a neurotransmitter associated with reward-related behavior and motivation. Dopamine pathways help reinforce actions that lead to beneficial outcomes, encouraging organisms to repeat behaviors that support survival. Another important neurotransmitter is serotonin. Serotonin plays a role in regulating mood, sleep patterns, and emotional stability. Changes in serotonin signaling have been linked to conditions such as depression and anxiety. A third example is norepinephrine, which influences alertness and the body's response to stress. Norepinephrine activity increases during situations requiring heightened attention and readiness. These neurotransmitter systems interact with limbic circuits to shape emotional experience. When dopamine pathways become active during rewarding experiences, they strengthen neural connections associated with those events. When serotonin levels shift, mood and emotional regulation may change. When norepinephrine rises during stress, attention and vigilance increase. Together, these chemical signals influence how emotional states develop and fluctuate. Yet emotions rarely arise from a single brain region acting alone. Instead, they emerge from interactions among many parts of the nervous system. Limbic structures communicate with regions of the cerebral cortex involved in reasoning and decision-making. Signals from the body also feed back into the brain, influencing emotional perception. For example, changes in heart rate, breathing patterns, and muscle tension can influence how emotions are experienced. The brain therefore operates as an integrated system in which thoughts, emotions, and bodily states interact continuously. A memory formed in the hippocampus may trigger emotional responses through the amygdala. Emotional signals may influence decision-making within the frontal regions of the brain. Neurotransmitters may shift the overall tone of mood and motivation. From these interacting systems arise the complex landscape of human thought and feeling. Although emotions may appear intangible or mysterious, they're grounded in neural circuits shaped by evolution and experience. Within the quiet activity of the nervous system, electrical impulses and chemical signals flow through networks that link memory, perception, and emotional meaning. Through these networks, the brain transforms sensory information and past experience into the feelings and motivations that guide behavior. The nervous system performs an extraordinary number of tasks every moment. It coordinates movement, interprets sensory information, regulates internal balance, stores memories, and shapes emotional responses. When functioning smoothly, these processes blend together so seamlessly that they rarely attract attention. Yet the importance of this delicate system becomes immediately clear when something begins to go wrong. Neurological disorders provide powerful reminders of how much everyday life depends on the proper functioning of neural circuits. By studying these conditions, scientists have learned a great deal about how the nervous system normally operates. One of the most well-known neurological disorders is Parkinson's disease. Parkinson's disease primarily affects movement. Individuals with this condition often develop symptoms such as tremors, slowed movement, muscle stiffness, and difficulties with balance and coordination. These symptoms emerge gradually as specific populations of neurons within the brain begin to degenerate. At the center of this process lies a small but crucial region of the brain called the substantia nigra. The substantia nigra is located deep within the midbrain and forms part of a network of structures involved in controlling movement. Neurons within this region produce the neurotransmitter dopamine, which plays a vital role in regulating motor activity. Dopamine acts as a chemical signal that helps coordinate the activity of neural circuits linking the basal ganglia, the motor cortex, and other parts of the brain involved in planning and executing movement. When dopamine-producing neurons in the substantia nigra begin to die, the balance of signals within these motor circuits changes dramatically. Without sufficient dopamine, the neural pathways that facilitate smooth movement become disrupted. As a result, movements may become slower and more difficult to initiate. Muscles may feel rigid or resistant to motion. Tremors can appear, especially when the body's at rest. Although Parkinson's disease presents significant challenges, the study of this condition has provided valuable insight into how motor circuits function in the healthy brain. For example, research into dopamine signaling has helped scientists understand how the basal ganglia contribute to the initiation and coordination of voluntary movement. Treatments developed for Parkinson's disease, including medications that increase dopamine activity, have also revealed how sensitive neural circuits can be to chemical balance. Another neurological condition that has greatly influenced neuroscience research is Alzheimer's disease. Alzheimer's disease is a progressive disorder that affects memory, thinking, and behavior. It is one of the most common causes of dementia, particularly among older adults. The earlier symptoms often involve subtle memory difficulties. A person may begin to forget recent conversations or struggle to recall familiar names. As the disease progresses, more profound changes in cognition can occur, affecting reasoning, language, and daily functioning. At the cellular level, Alzheimer's disease involves gradual changes in neural tissue that disrupt communication between neurons. One of the key features of the disease is the loss of synaptic connectivity. Synapses are the junctions where neurons communicate with one another. Healthy brain function depends on the integrity of these connections. In Alzheimer's disease, many synapses begin to deteriorate over time as these connections weaken or disappear. The networks responsible for memory and cognition become less effective. Researchers have also identified characteristic protein accumulations within affected brain tissue. Two proteins known as beta amyloid and tau form abnormal structures that are commonly associated with the disease. Beta amyloid molecules can accumulate outside neurons, forming plaques that disrupt communication between cells. Tau proteins can accumulate inside neurons, forming tangled structures that interfere with the internal transport systems of the cell. Together, these changes gradually impair the ability of neurons to communicate and function normally. Although Alzheimer's disease remains a major medical challenge, studying its mechanisms has greatly expanded scientific understanding of how memory-related neural circuits operate. For example, research into synaptic loss has highlighted the importance of maintaining strong connections between neurons for healthy cognitive function. Investigations into protein aggregation have also deepened understanding of cellular maintenance processes within the brain. Another perspective on neurological disorders comes from careful observation of individual patients. Few scientists have illustrated this approach more vividly than the neurologist Oliver Sacks. Oliver Sacks was known for his detailed descriptions of unusual neurological conditions and the individuals who experienced them. Through his clinical work and writings, he explored how disruptions within the nervous system can dramatically alter perception, memory, and identity. In one well-known case described by Sacks, a patient developed a condition known as visual agnosia. Although the patient's eyes functioned normally and he could see shapes and colours, he was unable to recognize familiar objects or even the faces of people he knew. The difficulty did not lie in the eyes themselves, but in the brain's ability to interpret visual information. In another case, Sacks described patients who experienced profound disruptions in their sense of movement or body position. Damage to specific neural pathways prevented the brain from accurately monitoring the body's posture, making even simple movements difficult to coordinate. These observations revealed how specialized different neural circuits can be. When a particular pathway is disrupted, very specific aspects of perception or behavior may be affected, while others remain intact. This selective disruption provides valuable clues about how the brain organizes its functions. Through careful clinical observation, neurologists have been able to link particular symptoms with specific regions or networks within the nervous system. In this way, neurological disorders have become powerful tools for scientific discovery. Studying what happens when neural circuits falter often reveals how those circuits normally operate. When dopamine-producing neurons decline, scientists learn how dopamine contributes to movement. When synaptic connections deteriorate, researchers gain insight into the mechanisms of memory and cognition. Even unusual perceptual disorders can illuminate how the brain interprets sensory information. While these conditions highlight the vulnerabilities of the nervous system, they also reveal its remarkable complexity. The fact that specific disruptions can produce such precise changes in perception or behavior demonstrates how intricately the brain is organized. Each neural pathway contributes to the larger network that supports thought, movement, and awareness. Through the study of neurological disorders, scientists continue to deepen their understanding of the nervous system. Each discovery brings new insight into the delicate balance that allows billions of neurons to work together in harmony. And in that understanding lies the possibility of developing new treatments that may help restore or protect the extraordinary network that quietly runs human life. For most of human history, the inner workings of the brain remained largely hidden from direct observation. Scientists could study the anatomy of brain tissue after death, and they could observe the effects of injuries or diseases, but watching the living brain in action was far more difficult. The nervous system seemed almost like a sealed black box, quietly producing thought, memory, and behavior, while revealing very little about its internal processes. During the late 20th and early 21st centuries, this situation began to change dramatically. Advances in technology allowed researchers to observe the structure and activity of the brain in ways that would once have seemed impossible. Today, modern neuroscience relies on powerful imaging techniques that reveal how different parts of the brain function and interact. One of the most important of these technologies is magnetic resonance imaging. Magnetic resonance imaging, often shortened to the acronym MRI, allows scientists and physicians to create detailed images of the brain without surgery or radiation. The technique works by placing the body within a strong magnetic field and using carefully controlled pulses of radio energy. Atoms within the body, particularly hydrogen atoms present in water molecules, respond to this magnetic field in predictable ways. By measuring these responses, computers can reconstruct detailed images of internal tissues. With magnetic resonance imaging, researchers can examine the structure of the brain with remarkable clarity. Differences in tissue density, blood flow, and fluid distribution can all be visualized, allowing scientists to detect abnormalities and study brain anatomy in living individuals. A related technique known as functional magnetic resonance imaging provides an even more intriguing view. Functional magnetic resonance imaging measures subtle changes in blood flow within the brain that occur when different regions become active. When a group of neurons increases its activity, it consumes more oxygen and energy. In response, nearby blood vessels deliver additional oxygen-rich blood to that region. By detecting these changes in blood oxygen levels, functional magnetic resonance imaging allows scientists to observe which areas of the brain become active during particular tasks. Researchers can ask volunteers to perform activities such as reading words, solving puzzles, or listening to music while lying inside a magnetic resonance scanner. The resulting images reveal patterns of brain activity associated with these tasks. These techniques have helped scientists identify networks involved in language, memory, attention, and emotional processing. Although the images do not directly display individual neurons firing, They provide valuable insight into how large-scale brain regions coordinate their activity. While imaging technologies reveal broad patterns of activity, another approach allows scientists to investigate neural circuits with extraordinary precision. One of the most innovative methods developed in recent decades is called optogenetics. Optogenetics was pioneered by neuroscientist Karl Deisseroth and his collaborators. This technique combines genetic engineering with light-sensitive proteins to control the activity of specific neurons. In optogenetics experiments, scientists introduce genes that produce light-responsive proteins into particular populations of neurons. These proteins allow neurons to respond to flashes of light delivered through extremely thin optical fibers. When light of the appropriate wavelength reaches these neurons, the proteins open ion channels in the cell membrane. This causes the neurons to become either more active or less active, depending on the specific design of the experiment. By controlling neurons with light, researchers can observe how changes in neural activity influence behavior. For example, activating certain neural circuits may trigger movement, alter emotional responses, or change patterns of learning. Silencing those same circuits may produce very different outcomes. Optogenetics therefore provides a powerful tool for exploring how specific neural pathways contribute to complex behaviors. The development of this method illustrates how modern neuroscience increasingly blends multiple scientific disciplines. Genetics, optics, engineering, and computational analysis all play roles in studying the brain. Beyond individual experiments, scientists are also attempting to map the brain's vast network of connections on a much larger scale. One ambitious effort is the Human Brain Project in Europe. The Human Brain Project was launched as a major international research initiative aimed at understanding the structure and function of the human brain. Scientists involved in this project seek to integrate data from many sources, including neuroanatomy, physiology, and computer modeling. One goal of the project has been to develop detailed digital simulations of neural circuits using powerful computing systems. By combining experimental data with large-scale computer models, researchers hope to better understand how billions of neurons interact within complex networks. Across the Atlantic Ocean, a similar initiative emerged in the United States. The Brain Research through Advancing Innovative Neurotechnologies Initiative, commonly known as the BRAIN Initiative, was launched to accelerate the development of technologies for studying neural circuits. The BRAIN Initiative supports research aimed at recording the activity of large numbers of neurons simultaneously, developing new imaging methods, and creating tools for analyzing massive datasets generated by brain research. One of the central goals of these efforts is to map the brain's connectome. The connectome refers to the complete map of neural connections within the brain. Just as the genome represents the full set of genetic information in an organism, the connectome represents the network of connections linking neurons together. Mapping the connectome is an enormous challenge. The human brain contains tens of billions of neurons, each capable of forming thousands of connections with other neurons. The resulting network contains trillions of synaptic links. Understanding this network requires advanced microscopy, computational modeling, and data analysis on a scale that was unimaginable only a few decades ago. Yet progress continues. Researchers use high-resolution imaging techniques to reconstruct tiny segments of neural tissue, tracing the paths of individual neurons and their synaptic connections. Computer algorithms then help assemble these fragments into increasingly detailed maps of neural circuits. At the same time, large-scale brain imaging studies investigate how distant regions communicate during complex tasks. Patterns of synchronized activity reveal functional networks that span multiple areas of the brain. These networks help coordinate processes such as attention, memory retrieval, and decision-making. The effort to map the brain therefore unfolds across many levels of scale. At one level, scientists examine individual synapses and molecular mechanisms. At another level, they investigate the interactions of entire brain regions working together. Bringing these layers together requires collaboration across many scientific fields. Physics contributes imaging technologies and measurement techniques. Computer science provides algorithms capable of analyzing enormous data sets. Biology offers insight into cellular mechanisms and evolutionary history. Together, these disciplines form the foundation of modern neuroscience. Although the brain remains one of the most complex structures known to science, new tools continue to illuminate its inner workings. Each improvement in imaging, genetics, and computation reveals more of the intricate networks that support thought, perception, and behavior. The effort to map the brain is still unfolding, but the progress achieved in recent decades has transformed how scientists explore the nervous system. What was once hidden deep within the skull is gradually becoming visible through the combined power of physics, biology, and computing. And with each new discovery, the quiet system that runs human life becomes a little less mysterious. Even when the body appears completely still, the nervous system remains active. Muscles may relax, the eyes may close, and conscious awareness may fade into sleep. Yet deep within the brain, electrical signals continue to flow through intricate neural circuits. The nervous system never truly stops working. From the moment life begins until the moment it ends, networks of neurons remain active, maintaining essential processes that support survival. The heart continues to beat. Breathing adjusts to changing oxygen levels. Hormones are released in carefully timed patterns. Memories are reorganized and neural connections are refined. Sleep, far from being a period of inactivity, represents a highly organized state of neural activity. During sleep, the brain enters patterns of electrical rhythms that differ significantly from those observed during waking consciousness. These rhythms can be measured using a technique called electroencephalography, which records electrical signals generated by large populations of neurons. Electroencephalography reveals that sleep unfolds through a sequence of stages, each characterized by distinct patterns of neural activity. One stage involves slow, synchronized waves of electrical activity sweeping across large regions of the brain. These slow wave patterns are associated with deep sleep and are thought to play an important role in physical restoration and memory consolidation. Another stage involves rapid bursts of activity known as rapid eye movement sleep. During this phase, the eyes move quickly beneath closed eyelids and brain activity becomes more similar to waking patterns. Rapid eye movement sleep is strongly associated with vivid dreaming. These alternating stages form cycles that repeat several times throughout the night. Although the body lies quietly in bed, the brain continues to coordinate complex physiological processes during these cycles. Understanding the importance of sleep has become a major focus of modern neuroscience. One of the researchers who has contributed significantly to this field is the sleep scientist Matthew Walker. Matthew Walker has studied how sleep affects memory, emotional regulation, and overall brain health. Through laboratory studies examining sleep patterns and cognitive performance, his work has highlighted how essential adequate sleep is for proper neural functioning. Walker and other sleep researchers have shown that sleep helps strengthen newly formed memories. Experiences acquired during the day initially produce temporary patterns of neural activity in memory-related circuits. During sleep, particularly during deep slow-wave sleep, the brain appears to replay some of these patterns. This replay process helps stabilize and integrate memories into long-term storage. In addition to strengthening memory, sleep also appears to support emotional balance. Insufficient sleep can disrupt the regulation of emotional responses, making individuals more sensitive to stress and less able to manage challenging situations. This connection between sleep and emotional stability reflects the close interaction between sleep-related neural circuits and limbic structures involved in emotional processing. Yet sleep may serve another crucial function that remained largely unknown until recent years. The brain consumes a tremendous amount of energy while performing its tasks. As neurons remain active, they generate metabolic waste products that must be removed to maintain healthy cellular function. For many organs in the body, waste removal occurs through the lymphatic system, a network of vessels that help clear metabolic byproducts from tissues. For a long time, scientists were puzzled by the apparent absence of a comparable waste removal system in the brain. This mystery began to change when neuroscientist Maiken Nedergaard and her colleagues proposed the existence of what is now known as the glymphatic system. The glymphatic system refers to a network of fluid channels that appears to help remove waste products from the brain. The name combines the word glial, referring to support cells within the brain, with the word lymphatic, reflecting its functional similarity to the lymphatic system elsewhere in the body. According to research led by Nedergaard, cerebrospinal fluid flows through channels formed by glial cells during sleep. This fluid moves through brain tissue, washing away metabolic waste products that accumulate during waking hours. Among the substances removed through this process are proteins that can become harmful if they accumulate excessively. Studies suggest that the glymphatic system becomes particularly active during deep sleep. When the brain enters slow-wave sleep, the spaces between brain cells appear to expand slightly, allowing cerebrospinal fluid to circulate more freely. This circulation helps carry away waste products and maintain a healthy neural environment. Although research into the glymphatic system continues, the idea that sleep plays a vital role in clearing metabolic waste from the brain has attracted considerable attention. It offers one explanation for why prolonged sleep deprivation can impair cognitive function and increase vulnerability to neurological disorders. From this perspective, sleep is not simply a period of rest. It is a time when the brain performs essential maintenance tasks that support long-term neural health. While the body sleeps, the nervous system continues to coordinate rhythms that regulate metabolism, hormone release, and immune activity. Throughout the night, neural circuits within the hypothalamus and brainstem help regulate the cycles of sleep and wakefulness. These rhythms are influenced by the body's internal biological clock, often referred to as the circadian rhythm. The circadian rhythm operates on an approximately 24-hour cycle and is influenced by environmental signals such as light and darkness. Specialized cells in the retina detect changes in light levels and transmit this information to a region of the brain known as the suprachiasmatic nucleus. The suprachiasmatic nucleus acts as a central timing system that helps synchronize physiological processes with the day-night cycle. When evening approaches and light levels decline, signals from the suprachiasmatic nucleus influence the release of a hormone called melatonin from the pineal gland. Melatonin helps signal to the body that it is time to prepare for sleep. As night progresses, neural circuits gradually shift the brain through the different stages of sleep. Through this orchestration of rhythms, the nervous system ensures that sleep occurs in a structured and beneficial way. Even though consciousness temporarily fades, neural networks continue their quiet work. Memories are reorganized, waste products are cleared, hormonal signals adjust to prepare the body for the coming day. In this sense, the nervous system truly never sleeps. Its activity changes rhythm and pattern during the night, but it never ceases entirely. Every moment, signals continue to pass through networks of neurons maintaining the processes that support life. Breathing continues, the heart beats steadily, internal balance is preserved. Even in the deepest sleep, the nervous system remains a vigilant guardian of the body's internal world. It operates quietly and continuously, sustaining the delicate balance that allows life to unfold moment by moment. For most of human history, the workings of the nervous system remained hidden from view. The brain and its surrounding network of nerves performed their tasks silently, shaping perception, movement, memory, and emotion, while revealing almost nothing about how these processes unfolded. The body appeared to respond to the world with remarkable precision, yet the underlying machinery responsible for that coordination remained mysterious. Over the past few centuries, however, scientific discovery has gradually illuminated the invisible system that makes human life possible. The story begins with the realization that living tissue can carry electrical signals. During the 18th century, the Italian physician Luigi Galvani performed a series of experiments that would reshape the scientific understanding of biology. While studying frog legs in his laboratory, Galvani observed that the muscles could twitch when exposed to electrical sparks. This observation led him to propose the idea of animal electricity, suggesting that living organisms possess their own electrical forces capable of driving muscle activity. Although Galvani's interpretation was debated by his contemporary Alessandro Volta, the experiment sparked a new scientific field that would eventually become electrophysiology. For the first time, researchers began to suspect that electrical processes might play a central role in biological function. The exploration of these electrical phenomena continued throughout the 19th century. Yet understanding how electrical signals travelled through the nervous system required another crucial insight. This insight emerged through the work of the Spanish neuroscientist Santiago Ramón y Cajal. Using improved staining techniques and careful microscopic observation, Cajal studied the structure of nervous tissue in extraordinary detail. At the time, many scientists believed that the brain formed a continuous network of fused fibers. Cajal's observations revealed a different picture. He demonstrated that the nervous system is composed of individual cells known as neurons, each communicating with others through specialized connections. This principle became known as the neuron doctrine. It established the idea that the nervous system consists of discrete cells forming communication networks rather than a single continuous structure. Cajal's work laid the foundation for modern neuroscience by revealing the fundamental architecture of neural circuits. Yet understanding the structure of neurons was only the beginning. Scientists still needed to uncover how electrical signals travelled through these cells. This challenge was addressed through groundbreaking research conducted during the 20th century by Alan Hodgkin and Andrew Huxley. Working with giant nerve fibers found in squid, Hodgkin and Huxley performed meticulous experiments that revealed how electrical impulses propagate along neurons. They discovered that nerve signals arise from the controlled movement of ions across the cell membrane. Specialized proteins known as ion channels regulate the flow of charged particles such as sodium and potassium. When these channels open and close in precise sequences, they generate the rapid electrical pulses known as action potentials. Through careful measurement and mathematical modeling, Hodgkin and Huxley demonstrated how these ionic movements produce the electrical signals that allow neurons to communicate. Their work revealed that the language of the nervous system is electrical at its core, governed by the laws of physics and chemistry. From these early discoveries, a remarkable picture of the nervous system has gradually emerged. Today, scientists understand that the human brain contains tens of billions of neurons, each capable of forming thousands of connections with other neurons. These connections, known as synapses, create a vast network of communication pathways linking nearly every part of the body. Within this network, electrical impulses travel along axons, while chemical signals cross synapses, transmitting information from one neuron to the next. Through these interactions, neural circuits coordinate an extraordinary range of activities. Sensory receptors detect light, sound, pressure, and temperature, converting physical energy from the environment into neural signals. These signals travel through intricate pathways to the brain, where patterns of activity are interpreted as sight, sound, touch, and other sensations. Motor circuits send commands outward from the brain and spinal cord, directing muscles to contract and produce movement. Autonomic pathways regulate the heart, lungs, digestive organs, and countless other internal processes without requiring conscious attention. At the same time, neural networks within the brain itself give rise to memory, emotion, learning, and thought. Each of these functions depends on the coordinated activity of billions of neurons communicating across vast networks. The scale of this system is almost impossible to fully imagine. Every moment, electrical impulses travel along neural pathways at remarkable speeds, linking distant regions of the body with the brain. Within fractions of a second, sensory signals from the skin may influence muscle movements. Emotional circuits may shape decision-making. Memories stored across neural networks may guide behavior in subtle ways. Despite this complexity, the nervous system operates quietly and continuously. Most of the processes that sustain life occur without conscious awareness. The body maintains its internal balance, adjusts its responses to the environment, and refines its neural circuits through learning and experience. Even during sleep, neural rhythms continue to organize memory, regulate physiology, and maintain the delicate balance required for healthy brain function. The nervous system therefore represents one of the most sophisticated biological systems known to science. It integrates electrical signaling, chemical communication, cellular networks, and evolutionary adaptations that have developed over hundreds of millions of years. From the earliest nerve nets of simple organisms to the intricate neural networks of the human brain, evolution has gradually shaped systems capable of sensing the environment, coordinating movement, and supporting increasingly complex behavior. In humans, this evolutionary history has produced a nervous system capable of language, creativity, reflection, and self-awareness. Yet beneath these remarkable abilities lies the same fundamental principle, first glimpsed in Galvani's experiments and clarified through generations of scientific discovery. Life depends on communication within living tissue. Electrical signals moving through networks of neurons link the brain to every corner of the body. Chemical messengers regulate activity across synapses and organs. Neural circuits integrate information, guiding behavior and maintaining stability. Through these interconnected systems, the nervous system orchestrates the countless processes that allow a human life to unfold. The sensation of warmth on the skin, the rhythm of breathing, the recall of a memory, and the quiet awareness of the surrounding world all arise from the coordinated activity of this invisible network. Although the nervous system rarely announces its presence, it quietly sustains every moment of experience. It is a silent intelligence embedded within the body, continuously monitoring, adjusting, and guiding the processes that make life possible. From the first electrical impulse in a sensory receptor to the complex patterns of activity that underlie thought itself, the nervous system operates ceaselessly in the background. And through its quiet work, the human body remains connected to the world moment by moment throughout the entire course of life.